Hello students, welcome to Vedantu NEET English. This is Miss Gopika, your own biology master teacher. Welcome all of you to our Navratri Series Day 4. So are you all super duper excited for Navratri Series Day 4? And today, for a change, I am going to be doing a little bit of botany. So happy Navratri to all the students who are celebrating, all the students who are not celebrating. And I hope all of you are excited because today is a Sunday and today we have a class as well, right? Yes. So show me your attendance in the chat box and also put a lot of fire emojis in the chat box. Let's build the energy up. Okay? Yes, I'm going to check if I'm audible, clearly visible also. Just give me a second. And, oh, is it fine now? So show me your attendance. Yes, very nice. Okay. Yes, we are clear, right? Everything is perfect? Chalo. Hello, hello. Yes, students, keep marking your attendance as well. Okay, keep marking your attendance as well. Okay. Yes, thank you so much. So let's start the class. So are you all excited for, I think many of you have already, been a part of my sexual reproduction and flowering plants class, but still, okay, for some people it might be new. How many of you are attending for the first time? Raise your hands in the chat. Okay, so students, sexual reproduction in flowering plants. Okay, this is one of the most beautiful chapters. At the same time, it fetches you around 3 questions. Okay, so it is going to fetch you, last year 3 questions were there, maximum was 5 questions. That means it's a very good potential chapter, no, that can go up to, giving you around 20 marks. Right, and it is very important for you to understand why is plants doing this. It's not just that you're giving rose for Valentine's Day. You should know why did the plant make rose, why did the plant even make that flower, what was the necessity for the plant to make the flower. Is it for you to cut it and give it to someone on Valentine's Day? Of course not. Plant doesn't even know you exist. So it is for something else, right? It is a change. It is because of an hormonal change, it's because of a morphological change that the plant has tried taking, right? Okay, thank you so much, students. Thank you so much. I want all the attendance to be marked, and I think many of them are missing today. Very lazy people. I'll expect them later in the class. So students, today we are going to first try to decode what is the word. Sexual reproduction in flowering plants is the word, right? Now tell me, what do you understand from this? Okay, what do you understand from this? Yes, 9 days Lahari. So students, look here. Sexual reproduction, that means what? Sexual reproduction means, ma'am, some variation is going to happen or some modifications are going to happen that is going to help the plant to do something better, right? Something better. Thank you so much, Hasini. So sexual reproduction means there is high chance for variation, right? There is high chance for variation. That is why we talk about sexual reproduction. Then what about the word flowering plants? Now in plant kingdom, all of you have learned about algae. Tell me about algae, bryophyte, pteridophyte, Gymnosperm and Angiosperm. Yes or no? Gymnosperm and Angiosperm. Tanuja and Madhumati, you can put the target. Okay, we have target for every class. You cannot escape the target. Okay, so algae, bryophyte, pteridophyte, Gymnosperm and Angiosperm. Yes or no? You have studied this in right now. I think you would have studied till Gymnosperm, right? Because Angiosperm is deleted. In this, all this, we know that every plant will go through a sexual phase of their life. There will be an asexual phase, there will be a sexual phase, right? Now, when the plant goes through a sexual phase, it is not necessary that the plant is going to flower, right? So, in Gymnosperm, the last thing that you studied in Gymnosperm was that, ma'am, they are going to bear naked seeds. Yes or no? They are going to bear naked seeds. So, if they are going to bear naked seeds, then we are not talking about Gymnosperm, we are not talking about pteridophyte, we are not talking about bryophytes, we are not talking about algae. About whom are we talking in this chapter? We are totally focusing on only one person, that is your Angiosperm, who is our flowering plant. Okay, who is our flowering plant. So here we are basically talking about our flowering plant. Is it clear? Yes or no? The flowering plant is none other than our angiosperm. Any doubts in the introduction, students? Any doubts in the introduction? Yes. Hello, hello. It's okay, it's okay. I think some of you will keep joining because some of them might think the class is at 6 o'clock also. They might not have checked the telegram. Okay. So the term is clear. We are learning sexual reproduction in flowering plants, and we are talking about who? Our angiosperm, who is more modified, and who is capable of giving us ultimately a result. What is the ultimate result that this angiosperm is going to give? It is going to form ovules. From ovules, we are going to get the seed, right? From ovary, we are going to get the fruit. So, students, this is why the plants are making the flowers, okay? Not for you to give it on Valentine's Day. Understood? Yes or no? Got it? Chalo. Chalo. Awesome. Awesome. Thank you. Thank you. Okay. All of you quickly mark the attendance, okay? Keep liking. Likes should keep increasing, okay? That's how the energy for Navratri series will be shown. Yes, ma'am, we understood the introduction. Now, students, let's go through NCERT few sentences that has been stressed in the, the paragraphs. Now, so plants reproducing is giving us a lot of emotions, right? Like if you are happy, you would give flower to someone. When they are getting married, right, they will have that garland flower. Again, when a person passes away, we give that for our expressing our feelings. Like that, when we are in when we are at a very good time of our life, or we have got a promotion, or we are at a very good time of our career, you will give flowers. So flowers are an indication, or it is related with lot of emotions. So that is what this paragraph is talking about. But one thing is there that is called as aids of sexual reproduction. Like just because plant is doing sexual reproduction, what is it benefiting? Okay, what is it benefiting? So look here, as an aid of sexual reproduction, flowers do not exist only for us to be used, not just for us, okay, but they are going to help in all kind of helping in what and all things, students. See, just because sexual reproduction is happening, we are able to see the flowers which are colorful. Yes or no? Flowers which are colorful, and not only that, we'll be able to see large flowers. Otherwise, you tell me, singing this butterfly song, will the butterfly be able to spot the flower? If the flower is so scutty like this, only this much is there, can you spot the flower? No. We need the flower to be bloomed, large, bright colored, so the insect can come and do pollination, right? Pollination. Got it? Awesome. Now, large flowers we'll get, and not only that, students, how many beautiful flowers are there that we use to make perfume nowadays? We get scents out of it, and also it also provides nectar as a reward for the insects. So, don't you think these are all the benefits of plants doing sexual reproduction? Agree, disagree, okay? Butterfly, where are you going? If you keep singing that, the butterfly will not be able to find the flower. Flower has to tell, hey, I am here. Look, beautifully standing for you here. I'm pink in color. I am broad. I am, I'm blooming. All that should be there, right? So that is very important. Now, a very important sentence here is, all flowering plants show sexual reproduction, okay? Like how we learned there, no? All vertebrates are chordates, but all chordates are not vertebrates. Like that, you have to know an important sentence, that is, all flowering plants show sexual reproduction. Got it? Yes? Okay. Now, a look at the diversity of structures of inflorescence. Now, what is inflorescence, students? Now, sometimes what happen, no? Even in class we would have noticed, no, that one student will be there. He is a potential problematic fellow, okay, one fellow in the class. Now with him there will be 2-3 people. Automatically they also become problematic. That means what? A group of people who start behaving like as one. Same way here, what happens? One flower cannot potentially be enough to attract an insect. So what these flowers are going to do, they'll be like, what, we are very small. So what we will do, we'll make an inflorescence. We'll make like a bunch. So when we make a bunch, okay, when we make an entire bunch, what happens? Insects will be able to see us. Yes or no? Agreed or not? One person cannot be a potential, problematic fellow. So what he will do, he'll make a gang of them. Okay. So inflorescence means what? A group of flowers, right? It can be cymose inflorescence and racemose inflorescence, right? I'm not going to teach this a lot because again morphology and your plant kingdom, you have learned most of the words, okay? This is not a new chapter for you, this is just a connection for you, okay? Connection for you. So, cymose is there, racemose inflorescence is there, that means a group, okay? A group. Now, inflorescence are there, flowers and their floral parts shows an amazing range of adaptations to ensure formation of the end product of sexual reproduction. What is our end product of sexual reproduction? Our ovule will become seed, ovary will become fruit, and we are going to eat that fruit, right? And it's not necessary that we consume all the fruits as well. Now, that's when I remembered I had bought some flowers, okay? That's when I remembered, I think all my flowers have become chutney, students, okay? Because I bought it from a little far, yeah. So, look at their condition, they look very bad. Okay, I bought it from very far, so they have all got into a very bad situation. Okay, this is the flowers. Morning I had sent you on Telegram. No, I don't think you can see. Okay, this. Can you see this flower? It's very. See this flower. Okay, this is an epipetalous condition. That means your stamens are attached to the petal. Okay, like you see in your Solanaceae family. No, this is attached actually. Okay, epipetalous condition it is. Yes. So ready? Okay. Yeah. 1 second, students. 1 second, 1 second. Yeah. Now, look here. Sexual reproduction, that is the fruits and the seeds. As I told you, this is our end product, right? Now, enzyme plus substrate gives us a product, means what? Okay, some enzyme plus substrate gives us a product means what? That means at the end of this reaction, I'm going to get a product, right? I'm going to get a product, okay? Now, in this chapter, let us understand the morphology, structure and the processes of sexual reproduction. So, I am hoping that all of you know your morphology, all of you know your plant kingdom. So, this chapter is going to be a connection of all of it, okay? Got it? Now, let's continue. Now, human beings have had an intimate relationship with flowers, like I told you, whatever emotion it be, expressing your love, expressing sadness, expressing you want to congratulate someone, you want to wish them good, all these times you are going to give them a bunch of flowers to showcase what you're feeling, right? So, we have a very intimate connection with flowers, got it? Very intimate connection, and not only that, in religious activities, right? Like now marriages also, we make sure that the entire decoration goes with flowers, okay? So, all that feelings, all of that is here. Now, see students, I know, you know, all of you cannot process that ma'am giving flowers to someone, but you have to learn about it, okay? Doesn't matter you belong to the single category or not, you have to learn a lot about flowers here, not just for like, you know, just to express, okay? You have to learn more than that, got it? Now, chalo, listen here. To a biologist, this is a very, very important thing. To a biologist, now let me tell you, a math student is there, a commerce student is there, art student is there. Now, if you show them, okay, if you show them a flower, then what will they tell? It's a flower. But if I show you a flower, you will also tell if it's a flower. That means what's the difference between you and them? You have learned more about flower. You have learned about enzymes, okay, hormones. You have learned about how the morphology. So, to a biologist, what is happening? Flowers are morphological and embryological marvels, understood? And also the site of sexual reproduction. So, for what other people, okay, like other people, if I ask you, okay, that students, I showed this and I'm like, what is this? Please identify. All of you will be telling, ma'am, flower, ma'am. You think I cannot see flower? It is flower, okay? But next time, what are you supposed to tell? You are supposed to tell me, ma'am, it is an embryological and a morphological marvel. That means this flower is able or is having the ability to do so many things, right? These flowers are ultimately going to form the seed and the fruit. How? Microsporogenesis is happening, Megasporogenesis is happening, double fertilization is happening, triple fusion is happening, right? So, not just being a Marvel fan is important, you should know the botany part of the Marvel. This also, got it? I don't know how many of you have even read this. Okay, first time they'll be thinking, ma'am, my god, what an information. Yes. Oh, nice, nice. Some of my old students are here, very nice. Okay, students, how much more for the target? Madhumati is here, Tanuja is here, that is our class leaders, okay? Yes. Chalo, now, look here, how are flowers formed? Have you ever thought about it? How flowers are formed? You'll be like, hey ma'am, why will I think about it? Very simple, no students. There is root apical, there is shoot apical meristem. Now root apical meristem, because of that the root will be formed, all of that is going to happen. Now shoot apical meristem is going to grow, and where are you going to find the flower? Tell me the answer. Are you going to find it in the root or the shoot? Okay, very easy question. Root or shoot? Root or shoot, where will you find the flower? If you do not answer this, we are going to have a war here. Okay, come on, jaldi tell. Shoot or root? Yes, okay. Let's see if we'll reach the target. It's obvious. There's I think there's a small delay in the chats. Okay, very good. Shoot. So students, I can say that these people, the plants, are actually reduced or modified shoots. No, not modified exactly, because there you're studying root modification, stem modification. So what is happening? A reduced shoot, right? So here we can say that these people, that is from your shoot apical meristem, okay, from your shoot apical meristem, the flower is going to be formed, yes or no? From the shoot apical meristem, which is potentially forming your shoot, right? That is getting a little condensed or modified. I'm not telling differentiated, but I'm telling modified, okay? Do not make the confusion. So when they modify, they form a bud. Definitely directly flower will not be formed, bud will be formed. Bud will be formed, okay? Or we can also say all the parts of the flower will be formed. From the bud, what is going to be formed? Flowers are going to be formed, right? Flowers are going to be formed. So from the shoot apical meristem, we are finally getting the flowers, right? Yes, we are finally getting the flowers. So this is how your flower will be formed. So students, with this you should also understand that, so as I told you, to a botanist, okay, or let's say biologist, okay, to a biologist, what is flower? In the chats, all of you tell me, what is flower to a biologist? First thing, it is a morphological, it's a morphological marvel, okay, morphological marvel, and it is an embryological marvel. And also one more thing, it's a site of sexual reproduction, right? Where the sexual reproduction is going to happen, okay? Wow. Ratis and you people know very skillful you are. You type green and you put blue color hearts. You put blue and you put red color hearts. Wow. What a color sense these people have, no? My students only. Should be proud. Okay, it's also a site for sexual reproduction. Site for sexual reproduction. Correct? This is what it is for a biologist, okay, botanist I'm telling, biologist, okay, that flowers are an indication to the morphology, embryology, as well as it's a site for sexual reproduction. Any doubts in the students? Now flowers will be formed, students, obviously there is different, role of your hormones coming here, like your auxin, cytokinin, as well as your ethylene, all these people are going to promote, all of that is going to happen, which you have already studied in your morphology. So I'm not going to explain that, but just understand this shoot apical meristem. Now one thing is, if a plant has decided, like now if you have decided to crack NEET, you have already made a plan, yes or no? You have already made a plan and you have reached the target telling that okay, I have to succeed in this. Same way, if a plant has decided to make a flower, okay, always remember, if a plant has decided to make a flower, two things are going to happen, okay. First thing is going to be, students, there is going to be an hormonal changes, okay, hormonal changes. As I told you, one will be promoting, one will be initiating, right? Like you have ethylene, you have auxin, you have cytokinin. All these are also going to help in the initiation. Continue with this, okay? Yes. So here, they are going to help with hormonal changes. They are going to already decide the structural changes. That okay, we have to bear a flower and we are going to bear it on the shoot, okay. These two things would already be decided. Understood all of you? Now you have decided to come and attend this class. You have already made your plan that okay, 5:15 I'll join ma'am's class. I'll stay as long as ma'am finished teaching and I'm going to learn this much out of this chapter. Correct? This is already decided. Okay, this is already decided. So this is going to be the two things that you have to remember. Is this clear? Any doubts? So how flowers are formed? Very simple. With the help of shoot apical meristem, they modify to form buds. Buds will become flowers. So the decision of becoming the flower is already made. So they will do two changes. First is going to be hormonal changes. Second is going to be structural changes. And next thing is, to a biologist, how do they see a flower? Okay. So I'll write that. To a biologist, what is flower? Okay, or what does flower mean? Anything, okay, anything. What is flower? It is a morphological marvel, embryological marvel, and site for sexual reproduction. And all those notes will be available on Vedantu NEET English Telegram as well as on Biology by Gopika. So join the Telegram if you want to get any more notifications. Now students, let's start drawing the flower. Ready? All of you? Okay, now look here. We are going to draw the flower. So first thing, you hold the flower and you give it to someone else. What are you holding? You are holding the stalk or you are holding the pedicel. So first thing you will do is draw a stalk. Okay, draw a stalk. This is the stalk of the flower. After that, students, there is a portion where all the parts of the plant is going to come and stick. Right, all the parts are going to come and stick. What is that portion called? A swollen region, that is called your thalamus. Yes or no? That is called your thalamus. This is the portion, right, where all of it will come and stick. Okay, all of it will come and stick. We'll shade this also in green. Students, artistic diagrams we have to draw. Okay. Yes, okay, done. Chalo. Now in the tip, okay, in the end there are going to be two leafy structures, right? Leafy structures. Now my flower is gone very bad. Okay, there are going to be two ends, no? What are that called? Okay, they are called as your sepals. Okay, these two portions, which is actually was helping the bud to be protected. Now if you would have seen rose bud or any bud for that matter, students, you would have noticed that there is a green color structure covering them. Who is that covering? This fellow was covering. Your sepal was the fellow who was protecting the bud so that it does not get any harmed. Now who is going to be the next one? The most colorful, prettiest person. It's going to be your petal, right? It's going to be your petal. So you can have your petal like this. Okay. Yes, petal. Got it? Should I draw a better petal? Wait, students, I'll draw one petal, okay? Yes, fine? Yes, beautiful flower. Okay, yes, petal is done. Now students, what else do you see? You will be seeing the anthers, right? The male part, they will have like this lobes. Okay, male parts will have like this lobes. Again here, filament, anther, done. Okay, done. Anther is done. Now what is going to be in the middle? What is going to be middle? Okay, these people are telling monopetalis and all. Wow, wow. Okay, too much information you people know. That is going to be your stigma, style and ovary. Okay, stigma, style and, oh, my ovary doesn't have place here. Stigma, style and ovary. 1 second, students. Yes. Yes, ready? Stigma, style, ovary. Got it? Yes, landing platform. Exactly. Very good, very good. So, shall we start labeling now? Now that we drew the flower, now we have to start labeling. Okay. So, what did I tell you? What you hold and give it to someone? What is that? Stalk. You are holding on the stalk of the plant. So, this is going to be the stalk of the plant. Okay. It can also be called as the pedicel. Okay. Pedicel. This is where you are going to hold and give the flower. Please, okay, take this. Now, this portion, the swollen portion is going to be what? The thalamus. It's going to be the thalamus. Yes. where all the parts are going to come and stick. All the parts are going to come and stick, right? Then this portion is going to be called your sepal. Sepal. Done? Yes, ma'am, done. This is your petal, the most colorful part which is going to help in attracting the insects. Then this portion is called your anther, and this long slender filament structure, this is your filament. Okay, filament. Done? One side labeling done? This is 3rd standards class, okay, 3rd standard, yeah. Now, this is going to be your stigma, which is where your pollen is going to land, right? This is going to be style. Students, I am going to label this side also, okay. This is going to be style, and this is going to be the ovary. Now remember, in the ovary, ovule is going to be there, and that's where you learned your placentation. Depending on how your ovule is going to be attached, you have studied your marginal placentation, parietal placentation, your free central placentation, basal placentation, all of this you have studied, no? That means the ovule is going to attach, and how many locules are there, all this you have studied in placentation. So students, you should understand that major chunk you have studied in morphology. Okay, now you are going to continue. Got it? Chalo. Now students, this is what a flower is of. But is this only about flower? No ma'am, there is something more about flower. That is, these flowers have whorls. What do they have? Whorls. There are 4 whorls for the flowers, right? They are going to have 4 whorls. The whorls are going to be starting with your calyx. Okay, calyx. Second is going to be your corolla. Third is going to be Androecium, and last one is going to be Gynoecium. Okay, Gynoecium. What are these? These are the 4 whorls. Inside these whorls, units are there. Okay, inside these whorls, units are there. Correct. So, these units are going to make these whorls. Now, example, let's start with calyx. Okay, let's start with calyx. Students, calyx, who is going to be the part of calyx? Sepal, right? Sepal is going to make the whorls. Okay, the unit is going to be sepal. Corolla, petal, ma'am. Okay, now, Androecium, students, Androecium is the male part of the flower. What is it? Male part of the flower. Very good, very good. All of you are answering. It is the male part of the flower. And the unit of who is going to contribute for making Androecium is going to be your stamen. Is going to be who? Stamen. So, among this, who is going to make the stamen? Anther and filament together is going to be called the stamen. Clear? Is this clear? All of you? Chalo, very good. Now, Gynoecium on the other side, okay, Gynoecium is going to be the female part, okay, female part. And this female part, who is going to be the unit? The unit is going to be your carpel or pistil. Carpel or pistil. Is this clear? Yes? Now, there is something called as essential part and non-essential parts. Which color do I choose? Okay, essential parts and non-essential parts. Now, essential parts means what, students? The ones who are going to help in sexual reproduction. The one who is going to help in sexual reproduction is going to be the essential part. Yes, ma'am, it's obvious. Okay, you are helping us to learn, so you will become the teacher. Right? So, same way, essential part means the one who is going to help in your sexual reproduction. Who is going to be the essential part? Can you all guess? Can you all guess who is going to be the essential part? Chalo, all of you. By the time I'll select a color, you can guess. Faster. Very good. Thank you, Soni. Tell me, how much are we near to the target? Oh, we are so far from the target, huh? Why is the energy low? Very good, very good. Ma'am, essential part is going to be your Androecium. Okay. So, in some people tell Androecium. Okay. You can tell Androecium. Okay. And Gynoecium. Why are we calling this the essential part? For simple reason, because the male part is going to form the male gamete. The female part is going to form the egg. These two should fuse. Then only we are going to get the embryo. Right? We are going to get the embryo. Correct? Exactly. All of you. Fast, fast. Okay. So, Androecium and Gynoecium is going to be the essential part. Now, what does non-essential part mean? What does non-essential part mean? Non-essential part means these people are not going to be directly involved in sexual reproduction. Okay. Non-essential part means they are not going to be directly involved in sexual reproduction, but they are going to be helping. Like how? Now, as I told you, butterfly, butterfly, where are you going? I am going near the colorful petal. So, the petal has to attract the insect. So, petal is going to do the job of attracting. Only if it attracts, the next step of pollen transfer is going to happen, because these bees and these insects will have sticky fur. So, to that pollen grains are going to stick, and when they fly, they are going to, scatter it on the plants, right, on the flowers. Now, for the insect to come and attract or look at this, oh, what are you doing? Oh, it looks nice, okay. For that, the flower has to be beautiful. Flower has to be beautiful. So, that job is done by who? Your corolla. So, non-essential part means they are not directly involving in sexual reproduction. That means they are not going to make the male or the female gamete, but they are going to do the side role. Like students in movie and all, like Ramcharan will be acting. And then there will be a one side friend, no? Like one secondary hero. He'll be telling that, you know what, she did this, she saw you like this. So, that is the second role, okay, secondary role. So, non-essential is going to be the people who are going to indirectly help. That is going to be your calyx and corolla. Okay, I am writing the words here. You can just remember sepal and petal as well. Excuse me. Okay, done? Yes. Yes, 11th, 12th, all the lives are held here, dear. You have to join. Got it? Yes. Clear? Okay. This is your entire flower thing. Now, students, ultimately, what is this flower going to do? Flower is going to be the site of sexual reproduction. Yes or no? This is the site of sexual reproduction. Site of sexual reproduction. Okay, you're going to get this beautiful notes on Vedantu NEET English Telegram. So make sure that you download the notes and keep. Okay. Yes. Thank you, Chintu. Nice name. My name is Mintu. Your name is Chintu. Okay. Chalo. Flower is clear. All of you, flower is clear. Now, these portions we are going to study again in detail. But right now you have to understand essential part, non-essential part. Is this clear so far? Pakka? Units of words also understood? Yes or no? Yes, ma'am. Okay. Yes, let's go to the next one. Now, events in reproduction. That means, what are we going to learn exactly in this chapter? Or what is our main focus for this chapter? So, right, that we have to understand. So, students, this chapter is basically divided into three parts. The first part, okay, the first part is going to be your pre-fertilization. What is it going to be? Pre-fertilization. Pre-fertilization. That means, let's take an example of all of you going for a trip. Okay, let's say full Vedantu NEET English teachers with Vedantu NEET English students are going for a trip. Okay, where do you guys want to go? Let's take small trip. Okay, don't tell ma'am I want to go to America, Africa and all. I'll leave you there only income. Let's take a kuti trip. Okay, let's say we are going to Kodaikanal. Okay, or we are going to Ooty. Okay, these are the places that we go. No. Now, when we are going for the trip, before I'm telling, the pre-event is going to start. That I'm going to contact you, I'm going to contact your parents and I'm going to ask permission, can I take these kids? Will it be safe for me? Will they be sure that they will reach? Okay, all that I'll take permission from them, and that is going to be the pre-part of the event. That means we select a place, I contact your parents, I take permission, I get the number of students who are going to come for the trip. Got it? Who is going to come for the trip? Now, next event is going to be where we are actually in the trip. Okay, where we are actually in the trip. Then it is going to be post-trip. That means after you come back from trip, you'll be like, oh, how beautiful the trip was. You know what? Gopika Ma'am scolded us. Gopika Ma'am danced with us. Okay, Shreyas sir told us the joke about Pandu and Chamba. It was very nice. They cooked food for us. So that is going to be the rest of it, right? So like that, for everything in our life, there is going to be a starting part, there's going to be a middle part, and then there is going to be end. Same thing is going to happen here, students. Pre-fertilization means what? Pre-fertilization means the event is going to happen where we make the gametes. We have to make the gametes, for fertilization to happen. So here, basically, we are going to do two steps that is called as microsporogenesis, okay, and megasporogenesis, I've added NCERT snip also, okay, so do not worry, we will be doing both. Yeah. And then you will do something and then I'll also come into the movie, no? I'm not taking you people. Trip is cancelled, okay? Pre-fertilization only, pre-event only got cancelled for you people. Okay, so now look here. Microsporogenesis is going to give me what? They are going to form microspores. That microspores is ultimately going to give me what? The male gamete, right? Male gamete. Don't tell me pollen grain, student. That is male gametophyte. Okay, it's going to give me the male gamete. Now, megasporogenesis is going to give me the egg. And now, for these two people have formed. Now, they have to meet or not? They have to meet. No, making and keeping, what you will do? They have to meet. So, for that, the second part of the same pre-fertilization event is going to be your gamete transfer. Okay, gamete transfer. Okay, gamete transfer. I should have written little more top, no? It's okay, right? Yeah, gamete transfer. Now, this gamete transfer is going to happen with the help of pollination. It's going to happen with the help of pollination. Now, let's go to the next event. What is the next event, student? The next event is going to be fertilization. Okay, next event is going to be fertilization, where they are actually, okay, this is the first step. This is going to be the second step, where the meeting is going to happen, fusing is going to happen. Fertilization, okay, where fusion of gametes happen. Okay, fusion of gametes happen. Clear? Yes. Yeah. Now, the third step and the last step is going to be post-fertilization events. Post-fertilization events. What is post-fertilization events? That means after fertilization, what is the ultimate? We have to reach somewhere. No, Chumma, you will keep going or what? You have to reach somewhere. No, that is going to be the formation of your ovule to seed, okay, ovary to fruit, your embryo, formation of embryo, your triple fusion leading to the formation of endosperm. This is going to be the ultimate destination. So, without taking you for a trip, I have taken you for a trip. So, are you all happy? Yes, you can all appreciate me in the chat box for taking you for a trip. Okay, imaginary trip. Fusion means what, Meghna? You're making tea and tea, okay? You mix milk and coffee. Sorry, you're making coffee. You milk coffee and milk. What is that happening? Fusion. Fusion of two things means what? Okay, coming together of two things. Yes, ovule to seed, ovary to fruit. Clear? Yes. Okay, done. Done. Fine. Now, what else? Events of reproduction is clear. Now, with this knowledge, we have to start from microsporogenesis, but before that, we have to learn about the stamen, we have to learn about the anther. Okay, learn about the anther. Okay, chalo. Now, students, let's see what NCERT has to say. Is it visible for all of you? Yeah, it's visible. Much before the actual flower is seen on the plant, the decision that the plant, okay, the decision that the plant is going to flower has been taken. I told you, no, same thing. Before you write the actual exam, you have already decided that I have to start preparing. So, the decision has already been made from your side, right? So, same way. So, what is going to happen, students? Several hormonal as well as structural changes are going to be initiated, which lead to differentiation and further development of floral primordium. Okay, floral primordium. Yes, primordium means just a set of organs or a tissues, okay, just like that, for floral primordium. Okay, now inflorescence are formed which bear the floral buds and then the flowers. In the flowers, the male and the female reproductive structures, that is going to be the Androecium, Androecium and Gynoecium, will differentiate and develop. You would recollect that Androecium consists of a whorl of stamen. That means what? The unit that forms the Androecium is going to be your stamens. Okay, stamen. Now, representing the male reproductive organ and Gynoecium representing the female reproductive organ. Any doubts? How can you have doubts? Come on. Yes. This paragraph is clear. So, only important words I am underlining. You do not have to read a lot. Okay, you do not have to read a lot. Yes. Chalo. Now, Androecium. Tell me, what do you know about Androecium? Ma'am, you teach, ma'am. Why are you asking us question? That is what you people are thinking, no? That on a Sunday you are asking us to use brain, ma'am. Exactly, they are completely colorblind. Okay, there are the different types of colorblind. Like normally we say blue, green and all. No? These people are some other level of this one. Yes, what's the target? How much are we near the target? Okay, come on. Madhumati and Tanuja, more energy we need. Chalo, let's start. Main Androecium means it's the male reproductive portion. Okay, male reproductive part. Students, in the male reproductive part, who is there? The whorl is Androecium, but who is forming the unit? Who is forming the unit? Unit is formed by your one and only, okay, stamen. And stamen is made up of what? Stamen is made up of two portions. That is nothing but your anther, okay, and filament. Anther and filament. Clear? Yes, very good. Okay, anther and filament. Chalo, let's continue. Now, stamen, anther and filament is there. Shall we draw the anther and filament? So now students, look here. What I am drawing is going to be your anther. These are the two lobes of your anther, and this is going to be the stalk-like structure. Okay, stalk-like structure. Done? Okay, can you see the anther? This is how your anther is going to be. In this flower also anther is there. I'll try to remove and show. Wait. Okay. Can you all see the anther? Can you all see the anther? Yes? Focus camera, focus. Yes, right? So, if I remove this, this is one anther. Okay. That went missing, students. It's too tiny, no? That's why. Huh. See, this is what? Okay, this is what we are going to learn. It's so small. Can you see the what I'm holding? That is your filament. Okay, what I'm holding is your filament. So, let's come back here. Look here. First thing or few things that you have to know is this portion. Okay, this portion is going to be your filament. Okay, this is your filament, and this filament is what? It is long, slender. It's long and slender and it's a stalk-like structure. Okay. Did he say that? Okay, focused, ma'am. Ma'am, zoom. If I zoom, then I have to set this all back. So, let's not do Katron ki Khiladi in class. Okay. Okay, let's not do that, students. Then you people will be very happy. Oh, class got ended. So, this is going to be bilobed. Yes or no? Two lobes are there. Students, this is going to be lobe 1. Okay, L1. This is going to be L2. So, this can be called as bilobed. Yes or no? This is bilobed. So, anther is bilobed. That means they have two lobes. Now, each lobe, okay, each lobe is going to have something called as theca. Okay, each lobe is going to have something called as theca. So this is one theca, this is one theca, this is one theca, this is one theca. So in one lobe, how many theca is going to be there, students? In one lobe, how many theca is going to be there? Yes? I know you people very well. That's why I didn't do any circus. Yes, very good. 2 theca, ma'am. In one lobe, there is going to be 2 theca. So we will call it dithecous. We will call it what? Dithecous. That means they have 2 theca. Done, done, done. Clear? Yes or no? Yes. 2 theca. Okay? Dithecous. Now, lobes understood, bilobe, dithecous. Now, entire your anther is, can I call it tetrasporangiate? Can I call it tetrasporangiate? Yes, ma'am. I will call this tetrasporangiate. Tetrasporangiate. So, students, if I check how many thecas do I have as a whole, okay, I'm just going to write this here. Just give me 1 minute, students. Yes. Dithecous, 2 theca. Tell me how many thecas will be there in the entire anther? How many thecas in an entire anther? It's going to be 4 theca, right? So we are going to use the word tetrasporangiate. Okay, tetrasporangiate. Very good. 4. Now, this is clear how the anther is. Anther is bilobe. 2 lobes are there. Each lobe 2 thecas are there. Okay, in a total 4 thecas are there, right? So they are going to have 2 plus 2 is equal to 4 thecas. Okay, 4 thecas, right? 1, 2, 3, 4. Now, if I do a cross section of this anther, okay, if I do a cross section of this anther, I am going to get a structure something like this, okay. I am going to get a structure something like this. Okay, I am going to get a structure like this. Got it? Now, inside the structure, students, you can just look here. Okay, same thing I have taken. I have just done a cross section like this. Okay, so this is going to have 1 theca. Okay, this is going to have 1 theca. This is going to have 1 theca, and this is going to have 1 theca. Yes or no? This is how it is going to be. Yes. What are you telling? No, you cannot say. No, no, no. Okay? Right. Thank you. Yes, they are tetragonal. So, students, look here. This is your, how, if I take a cross section, right, if I take a cross section, I am going to get a structure like this. Okay? Where these are going to be my lobes, right? These are going to be my lobes. So, if I draw here a dotted line, this is going to be my lobe, correct? This is going to be my lobe. So, 2 lobes. Sony. Very, very, what to tell, extraordinary students I have in my class. Very extraordinary, like when God cannot fight with them. That extraordinary these people are. Okay. Now, students, this is lobes. Clear or not? These are the lobes. Okay. This is bilobed. No doubt in that. Yes, ma'am. It is bilobed. So, what I am writing about your anther? You tell me one line about anther. It is bilobed. Okay. Bilobed. It is tetrasporangiate. Okay. Tetrasporangiate. It is dithecous. Yes, and it is tetragonal. Okay, and it is tetragonal. Got it? Yes or no? Yes, clear? Yes, okay. Oh, awesome, awesome. Now, if you look here, students, this is what is tetragonal, no? So, if I draw this like this, right, this is exactly how this formation is going to be, okay. Now, if I draw lines, let's see if I will be able to draw it, okay, one second. Shall I draw it with this green? Okay, purple I will draw like this, okay, like this, okay. It's actually thoda little difference is there, okay, tetragonal like this, okay, like this. It is going to be tetragonal like this, okay. Yes. So, like this, no, like this only 4 corners will be attached. So, this will be called as tetragonal, okay, this will be called as tetragonal. So, last point I am writing, they are tetragonal, okay. Now, students, in the middle I have drawn 2 lines. If you focused on it, I have drawn 2 green lines here, right. Here I am leaving some space and I have drawn some green lines. So, there let us say I am connecting with a green line here, okay, like this I am connecting with a green line here. Now, what is that green line? That green line is called line of dehiscence. Oh, we reached the target, okay, we should give the next target, okay. This line is called line of dehiscence. Line of dehiscence means what? The anther, after getting dehydrated, this will dehiscent, okay. This will dehiscent and this is going to release the pollen grains. Now, ma'am, you have drawn all of this. What is this circular structure? Yes, you understood they are thecas, but what are they? They are your microsporangium, okay, microsporangium. Got it? Okay, microsporangium. So, students, these are the microsporangium. So, when microspores are inside the microsporangium, we call it, if we call it microsporangium because microspores are inside it. When pollen is inside it, when pollen grains are going to be inside it, what will we call it? All of you in the chat box tell me. Yes, next target, exactly, okay. What is going to be, tell me, if pollen grains are inside it, what can I call it? Because microspores is inside it, I called it microsporangium. If pollen grains are inside it, what will I call it? Think and tell me. Yes. Not now, not anytime soon, okay. Now, next year only. Very good, Asha. Ma'am, you can call it pollen sac, right? You can call it pollen sac. So, right now, this is your microsporangium, okay. This is your microsporangium and inside this, what is there, students? Your microspores are there, okay. When they mature, when these people mature, they are going to be potentially going to be called, okay. If I label it here, students will think each one has one name. So, I am not going to do that. They will mature and they are going to form the pollen sac. Is this clear to all of you? Diagram is clear. We will draw an enlarged diagram, do not worry, but right now try to understand. Where did I take the cross section, students? I took the cross section here, okay. I took the cross section this way, okay. See, I took the cross section like this and I got this one, okay. I got this one, and if I would have done a cross section like this, I would have got one of the lobes like this, okay, one of the lobes like this. Is this clear to all of you? Yes. Now, let us draw the enlarged structure, okay, because we need to do the labeling and we need to do the paths. Ready, all of you? Energy is still there? Can I have some fire emojis in the chat? Okay, I want to know if all of you have the energy or are you tired? Is it okay? Yeah. Okay. So, students, look here. Now, I have drawn the structure. Okay, I am going to leave a little space for line of dehiscence. Okay, line of dehiscence, I have left the space. Now, we are going to learn some layers. Okay, we are going to learn some layers. Now, listen to me carefully. One layer will go. Okay, first I will just draw all the circles. Okay, I think it's too thick. 1 second. Yes, done. Magic. So many circles are coming. Okay, done. Okay, these are the 4 thekas. Okay, I think it's not looking that great. Okay, well, I'll just do back of this. Okay. Okay, 1 minute, students. Okay, here we have 1. Okay, here we have 1. I'm drawing a little bigger one, so I'll be able to show the shapes properly. Okay, here we have it. Done? Clear? Okay, these are the thekas, right? Okay, very good. Okay. No, it is not matured. Okay, I just stopped it at Microsporangium. When it becomes matured, we will draw the pollen sac. Okay, right now just listen here. Now, let's say when I am doing this, okay, when I am drawing this, okay, you have to this is so hard to get the same line measurement. Okay. Oh no, students, this is little, yeah, okay, we'll stop it here, even if it's perfect or not. Chalo, now this entire anther, right, these are the Microsporangium, you all understood that. Now each of this thekas or this Microsporangium has layers. They have 4 layers. So the first layer I have drawn, I am going to label this first layer as the epidermis. What am I going to label it as? Epidermis. Okay, first layer, epidermis. Second layer is going to be called the endothecium. So shall we draw the endothecium? Okay, shall we draw the endothecium? Second layer, okay, second layer is going to be the endothecium. So these 2 layers are single layers, okay, they are not going to be double. Okay, second layer, which you are seeing yellow color, I think it's not that visible. Okay, wait, I'll just draw it with another color. I think it should be visible now. Okay, this is going to be your endothecium. Yes, it's going to be endothecium. Second layer, the green one which I have drawn, is going to be your endothecium. Now next layer students is going to be multi-layered. Okay, it's not going to be single layer. First 2 layers were single layer, but the next layer is going to be multi-layer. So we are going to draw a thicker one. Okay, we are going to draw a thicker one. Okay, we'll draw a little thicker one. So we'll understand that this layer is having multiple other layers. Okay, so here we go. This is going to be your middle layer. Okay, this is going to be your middle layer. Clear? Orange color one, thick it is. Okay, thick it is. Yes, elephant ate my tomato. So this third layer is going to be called the middle layer. And the last layer is going to be called the tapetum. Okay, the last layer is going to be called the tapetum. So here we go, and we are going to draw the tapetum. So here in between they are going to have cells. 1 second students, I will just finish drawing this and I will explain to all of you. Okay, this is your tapetum. This tapetum students, it is a very nourishing layer, right? It provides the nourishment for the growing pollen grains. Okay, this is going to be your tapetum. Is it clear? Right? Okay, chalo. Now, elephant eats my tomato. I'll tell you what is the how to remember this. Let's finish labeling this. So this blue color one is your tapetum. Tapetum. Done. Now, ma'am, what are these people going to do? What is this epidermis, endothecium, middle layer going to do? Endothecium, middle layer, middle layer is going to do the job of protection. This is the exact line of NCERT. Okay. Protection and dehiscence. What are they going to do? Protection and dehiscence. Dehiscence means what, students? If the anther does not break, how will the pollen grains even go? Okay, if the anther does not break, right? If the anther does not break, the lobes, the microspores does not get scattered or the pollen grains does not get scattered, how will fertilization happen? So for that to happen, dehydration should happen, dehiscence should happen. That's why I told you there is a line of dehiscence. Okay, here I did not draw. So there is a line of dehiscence. Understood? This is going to be your line of dehiscence. So the breaking has to happen, right? The breaking has to happen. That is going to be done by your epidermis, endothecium and middle layer. Always remember, epidermis, endothecium is single layers, middle layer has 2 to 3 layers. Okay, middle layer has 2 to 3 layers. Now when we go to tapetum, we should understand that this layer is going to help with the nourishment. Okay, it's going to help with the nourishment, and it has dense protoplasm, because obviously if it has to provide nourishment, it should have a dense protoplasm, and it will have, it is multinucleated. It has more than one nucleus. Okay, it can be bi or multinucleated. Okay, multinucleated. Is this clear or not? Is this clear? Yes, all of you? All of you? Correct? Got it? Chalo, yes, very good. Tapetum provides nourishment. It is going to have dense cytoplasm, and it can be bi or multinucleated. Okay, bi or multinucleated, and these people are going to release the epidermis, endothecium and the middle layer. Now students, one more very important thing about tapetum is tapetum has an ability to release. Okay, tapetum also helps in formation of some substances. Okay, it's going to help in the synthesis of some substances like your callase, cellulose. So it is going to help in the synthesis of all of these materials, and these materials in the later classes you are going to understand where did we use this. Now students, as I told you, these are 2 lobes, right? This is 2 lobes. So if I draw a dotted line between these 2 lobes, okay, if I draw a dotted line between these lobes, don't you think we need to have a connection between these 2? Right, we should have a connection between these 2, otherwise how are they going to be together? So in between they have a connective. Okay, in between these 2 they have a connective. So I am just going to draw like this. Okay, this is the connective. To hold both the lobes together, there is going to be a connective. Any doubts so far? Any doubts so far? Just let me know. I think I'll do one thing, I'll just remove this green line. Might be confusing later for you people. Okay, let's say, okay, there is a connective. Okay, connective. Done? Clear? Sorted? All of you? I want all your answers, okay, not just 1 or 2%. I want all of you to tell me, was it clear or not? Okay, otherwise I'll think that, okay, only some understood, some did not understand. Clear? Yes. Okay. So the way to remember this, you have to remember this in order. Okay, epidermis, okay, epidermis, endothecium, middle layer and tapetum. Okay, this is called a connective, dear, which will connect both the lobes. Okay, this is a connective, I'll write it here, connects both the lobe. Okay, connects 2 lobes. Okay, connects 2 lobes. Now these, remember, these are your microsporangium. What are they? Right now they are your microsporangium, and what do they have inside them? What do they have inside them? Students, now when we, I'll tell you a joke, not a joke, I'll tell you a way to remember this. Now when we are in school, when we have, when we are in school and when we are stressed, what do we tell? Ma'am, I have study stress, right? I have stress from studies. Okay, one second. Yes, I have stress from studies. Same thing when we come to work or when we come to college, we'll tell we have college stress. Same thing when we come to work, we'll tell we have work stress. Then when you get married, you tell I have marriage stress, I have children stress, I have divorce stress, I have that stress, this stress, I have stress of retirement. So the stress words, right, you write the stress word and you just add the prefix, whatever it is, like I'm stressed with studies, I'm stressed with relationship, I'm stressed with, my teacher, I'm stressed with my friend. Stress is the same only, just that people keep changing it, whenever they feel like. Same way this is also. When microspores are inside it, we call it microsporangium. When pollen grains are inside it, we call it pollen sac. Same way our girl's pouch. Okay, first it was called as pouch. Okay, first it was called as stationary pouch. Then when we started putting lipstick and makeup in it, we call it makeup pouch. Okay, now we have tablets in it, okay, because that's the only thing we have to stress students. So we have tablet, headache tablet. So that will become our medical pouch. So we just, depending on what is inside the material, we keep changing the name of it. Okay, so this microsporangium, we are going to call it when it is, okay, when it is yes. When microspores are inside it, ma'am, you call it microsporangium. Okay, I'm calling it microsporangium because inside this there is going to be microspores. Okay, microspores are going to be inside this. Okay, now they tell that these microspores are formed because of, okay, microspores are formed because of a sporogenous tissue. Okay, sporogenous tissue. So students, technically if you are looking here, inside this it is going to be lined with sporogenous tissue. Okay, inside full, the middle portion is going to be completely your sporogenous tissue. This sporogenous tissue has the ability to undergo meiosis and form your microspore mother cell. Okay, microspore mother cell. Is this clear to all of you? Yes or no? Microspore mother cell. You will call it now the NEET pouch also, no? So microspores are there, and these microspores are formed from a set of tissues, and that tissues are called as sporogenous tissue. All this I am not making up, is there in your NCERT, okay? All these are lines of NCERT. So you understand this, then everything is going to be super duper easy, okay? Sporogenous tissue. Done? Done, done, done? Yes or no? Okay, now I told you about the trick. No, the trick is nothing but to order, remember this in order: epidermis, endothecium, middle layer, tapetum, right? So elephant eats my tomato, that is this one. Okay, you're a farmer. Okay, so you have tomato, you have a tomato farm, so that's what is happening. Okay, yes, hoping that all of you liked the session so far. Yes or no? All of you are enjoying, Pakka? Right, we are enjoying the session. Is there any student who is not enjoying? Right, got it. Okay, so this is clear, student. This is the enlarged structure that is given in your NCERT. I have also added the NCERT diagram. I will show it to you. Okay, I'll show it to you. But before that, just understand all of this. Okay, understand all of this. Again, once again, just remember 3 layers are there for your microsporangium. 4 layers are there: epidermis, endothecium, middle layer, and tapetum. In this, epidermis, endothecium, middle layer is going to help with protection and dehiscence. Okay, tapetum on the other hand is going to provide the nourishment for the growing pollen grains, as well as it is also going to have a dense protoplasm, and it is multi or binucleated, more than one nucleus. Okay, to keep both of the lobes together, we have a connective in between, and that connective is going to connect both of the lobes, because this is one lobe, that is one lobe, and each lobe will have 2 thecas, so it is called as dithecous condition. Now this is called as a microsporangium because microspores are inside it, and once these people mature, I will call it as the pollen sac. Okay, I will call it as the pollen sac because pollen grains are inside this. Is this clear? Yes or no? Chalo, okay, fine. Now we will go to NCERT. See, a typical angiosperm anther is going to be bilobed, okay, is going to be bilobed, and each lobe having 2 theca, that is they are dithecous. Okay, now often a longitudinal groove runs lengthwise separating the thecas. Let us understand the various types of tissues and their organization in a transfer session. So I told you, no, we did a transfer session to understand the lobes. We can also run a longitudinal session to understand about both the thecas. Okay, transfer session of an anther. The bilobed nature of the anther is very distinct in the transverse section of the anther. The anther is four-sided structure consisting of 4 microsporangia. Any doubts in this? 4 microsporangia located at the corners of each or 2 lobes. The microsporangia develops further and becomes the pollen sac. So that's why I told you before maturation, okay? Before maturation, you have to remember 2 words. That is microsporangium will have microspores. That's how the name Microsporogenesis, okay? Microsporogenesis. Now, same thing if it is after maturation, okay? After maturation, then what is it going to be, students? It is going to be called the pollen sac, okay? Pollen sac with pollen grains. See, I am teaching you NCERT plus I am making you write the notes. So, please be here, do not get distracted, okay? Yes, very, they are talking a lot, okay? Dehiscence means after dehydration, they will, see how will the pollen grains be exposed, how will the pollen grains come out? Only if the anther opens up. For that, we call this dehiscence, okay? Dehiscence, understood? So, yes, this is clear, students. So, the microsporangia develops further and becomes the pollen grain, okay? Becomes the pollen grain. Now, pollen sac, they extend longitudinally all throughout the length of the anther and are packed with pollen grains. Now, if you have seen your hibiscus plant, monoadelphous condition, students, you will know that there is a lot of yellow color pollen grains, right? Lot of yellow color, you will see dot. If you powder it and you see, it will be the pollen grains, okay? I had it, students, but the thing is, it has become so pathetic in condition that I cannot even show, okay? I dirtied my hand also. Okay, I bought the hibiscus, but the thing is, it's gone now. Okay, now look here, this is what I told you, there's a connective in between to connect both the lobes. This is going to be your first layer, that is your epidermis. Okay, this is a single layer, students, always remember this, it is a single layer, okay? Single layer, okay? Next one is going to be your endothecium. Again, this is also a single layer. Sporogenous tissue, as I told you, it will be placed in the center. This is a potential person who is going to form the pollen mother cell or your microspore mother cell, okay? Then you have last layer, that is your tapetum, and here you have the middle layer. This middle layer, the thing is, it is almost 3 to 4 layers, okay? It has 3 to 4 layers. Got it? Chalo, clear. Now, see, this is how it is drawn, same way even I do, to make you understand all of this. This microspore mother cell that they are talking about is what? This sporogenous tissue that is capable of forming the microspore mother cell. So far good? So far good? Clear? Okay, let's start microsporogenesis, all of you. Chalo, let's take a 2 seconds water break. All of you drink some water. All of you drink some water. Okay, chalo, let's start microsporogenesis. Now, what is microsporogenesis? Okay, we have to understand, no? 1 second. Okay, take 1 more second. Okay, chalo. Microsporogenesis means we have to form the male gamete. That is our job. Okay, we are doing the pre-fertilization event. Students, in this chapter, pre-fertilization event is little broader, rest everything is little small. Okay, so do not worry. Thank you, okay. Sony, I solved the issue. Okay, we solved the issue. This is also detailed only, dear. More detail I cannot go. This is the detailed. No. It's not Sunday. We took your leave forever. Okay, for a longer time we are going to take a leave for that. Okay, ads. Okay, ready? Now look here. we have to form the male gamete. And I told you, your sporogenous tissue, which was in the center, right, which was in the center, see, this was your sporangium, okay, I am not going to draw all of it, but yeah, this was the tissues, okay, this was your sporogenous tissues. Students, always learn with diagrams. I will tell you, visual representation is very important. You will always remember the diagrams more than what I was talking. Okay, that is how your brains work. That's why we remember movies more than reading a book. Yeah, this is your sporogenous tissue. Now, this sporogenous tissue in the middle has the ability to form, okay, sporogenous, okay, tissue. They are going to form what? They are going to form the microspore mother cell. They are going to form the microspore mother cell. This microspore mother cell, how is the division going to happen? How is the division going to happen? It is going to happen again from microspore mother cell with the help of meiosis. Okay, meiotic divisions are going to happen. With the help of meiosis, we are going to get microspore tetrad. What are we going to get? Microspore tetrad. 4 microspore tetrads. Okay, too small it became, no? Wait, students, I'll draw bigger one. Okay, fine. Still more big. Okay, I was just experimenting. Yes, okay. Yes, done. Now, look here. These people, okay, this is your microspore tetrad. This is your 4 microspore tetrads. But there is a problem. Problem is what you are going to see now. Okay, this is your microspore tetrad. This microspore tetrad, okay, this microspore tetrad is going to be covered with a layer. What is that layer? I have taught you people before. Tell me, let's see who will tell the layer. Okay, one whole layer, students, because of which they are not able to go anywhere. They are not able to go everywhere. Okay, they are like, I want to go there, I want to go here, but they are not able to do. Okay, tell me. Yes, very good. Yes. No, wrong, totally wrong. Students, this layer which is outside, okay, this layer which is outside is called, or it's formed of callus. What is it formed of? Callus. This callus makes sure that this microspore tetrad does not get separated. That means they'll all be together. So this microspore tetrad is going to have an issue. That issue is going to be that these people are not going to get separated. So what is going to happen? What is going to happen? Yes, very good, very good. Who is that? Udaya Prakash is the only one who told. That callus has to be removed or broken. If the callus is not broken, we will not get this microspore tetrad separately. Okay, microspore tetrads separately. Now, for that to happen, what we are going to do, students? We are going to take the help of tapetum, because tapetum has made or synthesized callus. Okay, callus. That's why I told you, remember this and keep, we are going to learn it later. This callus is going to break open this callus and we are going to get our microspore tetrads, free microspore tetrads. Understood? So, we are going to get 4 microspore tetrads. Is this clear to all of you? Yes or no? Got it? Now, each microspore tetrad is a potential pollen. That means each of them will become a pollen grain. So, let's just pick one of the fellows, okay, one of the microspores, and let's see what is going to happen. Students, they are going to form the pollen grains. They are going to form what? Pollen grains. So, this is going to be the intine, okay. Outside this, around this, there is going to be a structure. I think all of you know the structure, isn't it? Okay. It's your sporopollen, okay, sporopollen. So each microspore tetrad is capable of becoming a pollen grain. All of them have the potential ability to become a pollen grain, and this pollen grain will have two cells. Okay, one is called your generative cell and one is called your vegetative cell. So this your vegetative cell will have an irregularly placed nucleus, irregularly shaped nucleus, and this is going to be larger. Your vegetative cell, I think I'll draw a little more down for you. Okay, here. Okay. Your vegetative cell is going to be the larger cell. Okay, vegetative cell is going to be the larger. Because why? It has the food reserve to give it to the generative cell. So, to be frank, okay, why brain is not braining? I'll hit you people. Okay, to be frank students, you should never say that your pollen grain is the male gamete. Pollen grain is the male gametophyte because generative cell is going to have your male gamete. Any doubts? Any doubts? Tell me please. Yes, spindle shaped nucleus. Yes, we are going to come to that. Okay, we are going to come to that. So students, let's label what we have written. Let's just label what we have written. So first layer is going to be your intine. Okay, intine is made up of cellulose and pectin. Okay, it is made up of what? Cellulose and pectin. Students, this question has come. So you should know what these layers are made up of, cellulose and pectin, and let me tell you, this is a continuous layer. No breaking is there. Okay. Okay. So cellulose and pectin, it's a continuous layer. Continuous layer. Now on the top, what do we have? We have sporopollenin, which is one of the hardest substance. Okay. One of the hardest substance present, which cannot even be degraded with the help of an enzyme. Okay, an enzyme also is not able to degrade this. Why? Because it's one of the hardest materials. No enzymes are there, no acids are there, no alkalis are there, which is ever found who is going to degrade this. This is so resistant. So sporopollenin is a discontinuous layer. Why? Because can you see I have left a space here. This is called the germ pore, through which the pollen tube can arise. Because students, this material is so hard. So if the opening is not there, how will the pollen tube form? How will the male gamete go up to the egg? Transport system should be there, no? Transport system should be there, no? For the transport system only, we have to do this. Okay? Is this clear? Chalo. Now look here, this is the germ pore through which the pollen tube, okay, germination is going to happen. Pollen tube growth, I'll tell. Okay? Pollen tube growth. Then here sporopollenin, this is the hardest, okay, hard material. Now tell me about the cells, students. Two cells are there. First one is going to be called the vegetative cell. The large cell is the vegetative cell, okay, vegetative cell, and this is going to have dense cytoplasm. They are going to have the food reserve for the growing male gamete, that is the generative cell. Okay, generative cell is going to have the male gamete. Is this clear? So is pollen grain a gametophyte or a gamete? Tell me this, all of you in the chats. Pollen grain a gametophyte or a gamete? Faster. Your answer matters, okay? Your voice matters, we tell, no, like that. So spindle shaped nucleus, okay? The cell also is kind of spindle shaped only. Very good, Satvik. Who else told? Satvik told. Anusha, very good. Rani, very good. Okay, Yashu, Sneha. Yes, it is preserved as a fossil. Very correct. Okay, Pavitra, very good. It's a gametophyte. I can call, this is a pollen grain. Yes or no? This is a pollen grain. Inside the pollen grain, male gamete is there. Male gamete is going to be where, students? In the generative cell. So I cannot call this as a male gamete. I will call pollen grain as a male gametophyte, and I'll call the generative cell male gamete, who is going to have the male gamete. Understood? This question will come. That's why I'm stressing it again and again. They'll ask you who is a gamete, who is a gametophyte. Okay, who is a gamete, who is a gametophyte. Is this clear to all of you? Is microsporogenesis clear? Any doubts in microsporogenesis? Please let me know in the chats. Any doubt? These are the important things, so I am putting the boxes, okay, marking the boxes so you remember. Okay, my sporogenous tissue. So I will say pollen grain, okay, pollen grain is a male gametophyte, okay, male gametophyte. Now, a very, very important thing from this chapter, listen to me carefully, okay, 60%, okay, what did I tell? 60% of the angiosperm of the, or 60% of the flowering plants will dehiscent at 2-celled stage. At what cell stage? 2-celled stage. But rest 40%, okay, rest 40% will dehiscent at 3-celled stage. At 3-celled stage. Is this clear? What did I tell? That means this is the 2-cell stage. Generative cell, vegetative cell is there. Vegetative cell will give all the nourishment to the generative cell and tell that you please make the male gamete. I will sacrifice everything for you. 2-cell stage. Okay. That means later the male gamete will be formed when the pollen tube germination happens, and that will continue. 3-cell stage means, students, here 1 vegetative cell, 2 generative cell. That means 2 male gamete. 1 male gamete will go and fuse with the embryo, main fellow. 1 male gamete will go and fuse with the polar nuclei. So, 1 will form the embryo, 1 will form the endosperm. That is called as 3-cell stage. So, 60% of the angiosperm will go for a 2-cell stage and 40% will dehiscent in 3-cell stage. Is this clear to all of you? Yes or no? It's a very important question. So, please make a note. Please make a note. Okay. Happy Navratri, Chaitanya. Yes. Quickly tell me. Any doubts? Clear, all of you? Ma'am, is vegetative cell a somatic cell? No dear, the whole pollen itself is a gametic cell, no? That is going to form the male gamete. So, we cannot call it somatic cell, right? Vegetative cell in our organisms, that we call it as a somatic cell. But this as a whole is a gametic cell, okay? Clear? Perfect, okay? Satvik, clear? Doubt is clear, dear? Chalo, okay, fine. Yes, I am from Karnataka. Okay, microsporogenesis, pakka clear? No doubts? Everything is super clear? Okay. I was just thinking what to write. Do you guys want a flow chart? Do you guys want a flow chart? Because that slide is a little crowded. So, do you guys want a flow chart here? I can just write a flow chart here faster for all of you. Okay. So, look here. Okay. So, you have microspore mother cell. Okay. Microspore mother cell, which you call it as MMC. Okay, and this MMC will undergo meiosis. Students, here you will not get number of nuclear division questions, but you will get it in Megasporogenesis, because there nuclear divisions are happening. Okay, free nuclear divisions are happening, some cellular divisions are happening. So, their question can come. Here it will not come. Okay, here it will not come. Now, Meiosis will happen. This will form your microspore tetrad. Okay, microspore tetrad. And this microspore tetrad will become each microspore, okay, each microspore can become a pollen grain. Each microspore can become a pollen grain, and that means they are capable to be a potential male gametophyte. I am writing Mg, okay, don't think it is Morris Garrett, it is Mg, male gametophyte. Okay, male gametophyte. Now this pollen grains, inside that two cells are going to be there, vegetative cell and generative cell. In that generative cell is going to bear the pollen grains. I mean sorry, it's going to bear the male gamete. Male gamete. Clear? Yes or no? Perfect. Okay. Krishna, yeah, we will come to that, dear. Okay, we'll come to that, don't worry. We have questions on that, we also have NCERT questions on that, okay? Yes, don't even worry. Mitosis, again, we'll tell you mitosis when you are studying for your this one. Okay, chalo, clear. Do you guys want a break now or after this? I think we can finish this, no? Okay, see structure of microsporangium. In a transverse section of an anther, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers. Okay, what are the 4 wall layers? Epidermis, endothelium, middle layer and the tapetum. Okay, now the outer 3 walls perform the function of protection and dehiscence. Okay, and helps in dehiscence, as I told you, dehiscence. And to release the pollen. Release the pollen means if dehiscence does not happen, release will not happen. Okay. Now, the innermost wall layer is called the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possesses dense cytoplasm. Okay, cells possesses dense cytoplasm and generally have more than one nucleus. Okay, I told you, no, they have more than one nucleus. It can be binucleated or multinucleated. Now, when the anther is young, a group of compactly arranged homogeneously cells are called as sporogenous tissue, which occupies the center, center of the microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergoes meiotic division. So, students, here only meiosis we are talking about right now, to form microspore tetrads. Okay, microspore tetrads. What would be the ploidy of the cells of the tetrads? Can you guess and tell me what could be the ploidy? Just imagine. Okay, we have microspore tetrad. Okay, we have 4 microspore tetrads. What could be the ploidy, students? Just normally think 4 microspore tetrads are there. What could be the ploidy? Yes, this question I have answered before for all of you. And students, you know what? Okay. Sexual reproduction in flowering plants, short form SRIFP. It's too difficult to type the full name, no? Very good, very good. They can all be haploid, right? N, and N. They will be haploid. Very good, very good. Okay, chalo. Now, understood, no? The question is answered. Now, as each cell of the sporogenous tissue is capable of giving rise to microspore tetrad. Okay, microspore tetrad. Each one is a potential pollen or a microspore mother cell. As I told you, everyone has the potential to become the microspore mother cell. Okay, to form the formation of microspores from the pollen mother cells through meiosis is called as microsporogenesis. Clear, right? From sporogenous tissue, we can form the microspore mother cells. Microspore mother cells will undergo meiotic division and it will become the microspores. That microspores will become potential pollen grains. Okay. Dear, I finished all that. Okay, I already finished it previous topic. Okay, now the microspore, as they are formed, are arranged in clusters of 4 cells called the microspore tetrad, and they are arranged this way because of the presence of callose, which will keep them together. We have to break them open for them to become the pollen grains. Now, as the anthers mature and dehydrate, the microspore dissociate from each other and develop into the pollen grains. Okay. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with dehiscence of anther. Okay, dehiscence of anther. Clear or not? So far good? So far good? Microsporogenesis pakka clear? Yes or no? Any doubts? Please let me know. Jaldi, jaldi tell me. Any doubts? Okay. So students, here a question can come to you. See, look here, okay. Now up to this stage, okay, I'm just going to write few things here. See, microspore tetras, before this one meiosis happened, microspore happened, each microspore was going to become a pollen grain. So during this phase, okay, where your microspores will become the pollen grain, here mitotic divisions will happen. Okay, here mitosis will happen and they will form the pollen grains. Okay, just remember this. Got it? Clear or not? So that your confusion will not be there. One meiotic division would have happened and one mitotic division would have happened. Okay, one reduction division, one equational division would have happened. Yes. So your microspore mother cells should always be diploid, right? 2n it should be, and these people are going to be here. These people are going to be haploid. These people are going to be haploid. Students, now one thing I want to add on here near your anther is, students, as I told you, epipetalis condition, epitepalis condition, all this you would have studied in morphology. Okay, morphology, no? So, okay. So, here students, I want to tell you one small thing of your NCERT. Since this fellow, who is that? Your filament is going to be attached to the thalamus. Yes or no? Everything is going to be attached to the thalamus. Yes? This two lines have been given in your NCERT, and maybe in case a question comes, I don't want you people to lose even 1 mark. So, here students, see, you can see now this is attached to the thalamus. The end which is attached to the thalamus will be called as the proximal end of the filament. What end? Proximal end. That means the end which is very closer to the thalamus is going to be called the proximal end. Okay. Nagesh, there is a Kannada channel, you can search Vedantu, NCERT, Vedantu, KCET, Karnataka, you will get the channel, okay, there will be Kannada. Yes, this is going to be the proximal end, and the from the thalamus, this portion is very far, right, very far. This is going to be the distal end, okay, this is going to be the distal end. Any doubts in this? The end which is far is the distal end, the end which is close by is going to be the proximal end. Yes, very good. Okay, I just remembered it suddenly. So, when we teach, no, sometimes some portions we will skip by mistake. So, then later we will remember it. So, whenever we remember, we have to teach. Yes, ready? Okay, yes. Now, pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent two-layered wall, a hard outer layer called the exine, okay, which is made up of sporopollen, which is one of the most resistant organic matter known, which can also withstand high temperature, high acidity, high alkaline. It's like whatever you do, like sometimes your mom tells, no, how much ever I scold, you have no change. That means what? She has assumed you to be the hardest material in that house. Okay, whatever she is telling, even if she is throwing something, hitting you also, no movement has happened, no change has happened, or change has happened for 5 minutes. So, technically, you will become sporopollenin of the house. Got it? Okay, you will become sporopollenin of the house. So, exine is made up of sporopollenin. Okay, exine is made up of sporopollenin. Intine is made up of cellulose and pectin. Okay, now, nothing is there to degrade sporopollenin. So, the pollen grain, the exine has a prominent aperture called as the germ pore. Aperture means a small, depression. Got it? Okay, depression. Now, where sporopollenin is absent. Pollen grains are well preserved as fossil because of the presence of sporopollenin, because obviously nothing can degrade this fellow, no. The exine exhibit a fascinating array. Students, there are a lot of ornamental plants, no. There are a lot of shapes of pollen grains as well, a lot of shapes. If you look under the microscope, the normal hibiscus pollen grain also will look so beautiful, they will have a pattern. Okay, that pattern is, a lot of people learn about the patterns and they understand, okay, this is how this is and all of that, okay. Now, the inner wall of the pollen grain is called intine. It has a thin continuous layer made up of cellulose and pectin. Any doubts so far? Tell me, any doubts so far? And the cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell, okay, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserve and large irregularly shaped nucleus. The generative cell is small, floats in the cytoplasm of the vegetative cell and it is spindle shaped, okay, spindle shaped with a spindle shaped nucleus also, with dense cytoplasm and a nucleus. Dense cytoplasm and a nucleus. So far good? Okay. Now, in over, see this is the most important line, very very important. In over 60% of the angiosperms, pollen grains are shed. Okay, dehiscence is going to happen at the two cell stage. That means one generative cell and one vegetative cell. Okay. In other species, what is going to happen? Generative cell will divide mitotically to give rise to two male gametes before pollen grains are shed to a 3-cell stage. So here what can happen? From microspore, okay, from microspore to pollen grain, okay, one mitotic division would have happened. This is in the 2-cell stage. From the 2-cell stage to a 3-cell stage, one more mitotic division will happen. Got it? So students, you have to count how many mitotic division happened, okay? It's your job to count and think, okay, this many mitotic division has happened. They have told in this places. Got it? Yes. Clear? Okay. Before I go to this, you'll take a break. Okay, I think it's almost 2 hours. Okay, so we'll take a break. How long you want the break? Yes. Okay, break is given. Go and come back. Shouldn't run away, okay? No one should run away. Yes. So, bye-bye. See you guys soon. Yes. Ready? Welcome back. Oh, I think I should inform these people. Otherwise they would have gone. Yes, students, I'm just informing the students. Okay, chalo, done. Okay, so students, next topic is pollen grain allergy. Okay, now all of us do not have pollen grain allergy, right? Do all of us have pollen grain allergy? Yes or no? Voice is audible, no? No problem, right? I think no one has an issue. Okay, chalo, ready? Happy Navratri Sudarshan. Yeah. Okay. So students, pollen grain allergy means what? That means we have allergy from pollen. Simple as that. Okay. So an allergy or an inflammation caused due to pollen grains. Right? An allergy or an inflammation caused due to pollen grains. Simple. Right. Now, do all of us have allergy? No, ma'am. All of us do not have allergy. All of us have allergy with different things, right? For every single person, allergies will be different. Everyone will have different types of allergies. So, if you have an allergy with pollen and that causes you an inflammation, it could even lead to asthma. Okay, it can lead to asthma. It can lead to blocking of your bronchitis. Okay, bronchitis, etc. Okay, now which are the weeds, that is, which are the pollen grains which are harmful, or which are the plants? Yeah, dust allergy is there for all of us. Okay, which are the plants that are very causing us a lot of allergies? One of the examples given in your NCERT, you have to remember, that is Parthenium grass. Okay, Parthenium grass, also called as carrot grass. Okay, or called as carrot grass. This was actually a weed, students, which had come from imported wheat. Okay, this had got mixed with the imported wheat, and right now it has become a big trouble to all of us. Wherever a little empty space, this will start growing. It was first found in imported wheat. Understood? That's all. Nothing much about your allergy or anything. Okay. So, we cannot resist this kind of allergies. What we can do is, we can be away from such places, okay, where you see a lot of plants, or where there is a lot of weed, or someone gives you a bouquet of flowers. You can inform them that I have pollen allergy, please don't bring it near me. Okay, yes, clear? Chalo. Now, next topic that we have to learn is viability. Now, viability means what, students? How long after dehiscence this pollen is going to be active? Full time the pollen won't be active. No, now you people also, starting you will be fully active. Yes ma'am, we will like ma'am, we will comment ma'am, we will do that ma'am, we will share. Okay, we will make you Miss Universe, we will make you that, we will make you all this, you will tell. But does any of that happen? Nothing will happen. Okay, after one hour you will be like, ma'am, do you think we can complete this chapter, or should we do it in 2 parts, 3 parts? Okay, after 2 hours you'll be like, ma'am, how long? When will the session end? So, as and when your time period continues, you will get tired. Same way, as dehiscence happen, okay, when the anther is out, okay, dehiscence happen, how long this is going to be viable? How long can we use this? Just like, students, your OTPs, right? Like, once you use your debit card, credit card, you will get an OTP, and they will tell you in 2 minutes your OTP will expire, in 5 minutes your OTP will expire. What does that mean? How long this OTP is going to be valid? That is what it is. Okay, how long this OTP is going to be valid? That is what the viability is. So, viability of different plants is going to be different. First thing they have given in your example, in your NCERT, is rice and wheat. What is it? Rice and wheat. They have given that these people can be viable for few few minutes, that is up to 30 minutes. Okay, few minutes up to 30 minutes. Got it? After that, these people are of no use. Okay, these people are of no use. Got it? See, they are already asking, such monkeys. So, rice and wheat is going to be viable for 30 minutes, that is for few minutes. Okay, then you have Rosole. This is the trick to remember. RO for Rosaceae family. Okay. Solanaceae. Okay, and LE for Leguminosae. Okay, these three people, okay, these three families can be viable for a longer period of time. That means these people's pollen grains can be viable for few months. Okay, few months. Now imagine the viability is very less. Okay, very less. Then what to do, ma'am? How will I, I have to make sure that I am becoming one of the richest farmers. What am I going to do? I'll tell you, please do cryopreservation. What will you do, students? You will cry and preserve it. Okay, that is called cryopreservation. Not really. Okay, cryopreservation means you preserve the seeds in a temperature of minus 196 degrees Celsius in liquid nitrogen. Okay, in liquid nitrogen. Understood? If you cry and preserve, nothing is going to happen. After that, they are not going to be viable. Oh, target and wow. Today. Clear? Yes or no? Any doubts? Okay, you will keep it in liquid nitrogen. Okay. Okay, you will keep it in liquid nitrogen at a degree of minus 196 degrees Celsius in liquid nitrogen, and you will do cryopreservation. Just like your blood bank, okay, we will also have pollen bank. We will have what? Pollen bank. So, when we talk about viability, we also have to remember, students, viability also depends on, okay, these things also depends on the temperature. Depends on temperature, humidity, okay, and other environmental issues also will affect their viability. Okay, not just that, okay, I got released here, 30 minutes over, I will go and die. It's not like that. They will wait. Okay, environmental conditions changing, I have to change. So that is going to happen. Okay, yes. What happened to Sanjay? Don't be emotional now. Okay, what is happening? So, it depends on the temperature and the humidity. Is this clear? Viability is a very kutty topic, okay, but you have to remember these family names. Examples you have to remember, otherwise gone, okay, otherwise gone. Same thing is written here, students. You can see cryopreservations. So, what are the important lines here? See, we have to, how long do you think the pollen grains retain viability? So, here you can see in some cereals such as rice and wheat, okay, rice and wheat, pollen grains are going to lose their viability within 30 minutes, okay, of their release. Now, in other families like your Rosaceae, yes, in other ones like your Rosaceae, Leguminosae and Solanaceae, they maintain viability for months, okay. Then you may have heard about storing of semen, storing of eggs. Same way we have storing of pollen grains, which is called as cryopreservation. That is, in liquid nitrogen, we will keep it at minus 196 degrees Celsius. Any doubts? Any doubts? Tell me quickly if you have doubts. We are going to come to the next most important portion. Did I draw this line? Wait, this line was already there. What happened to the board, yeah? I hope nothing happens. Okay, chalo. Students, from when this is there? Now it started coming, no? Or before it was there? I didn't see only. They're telling me to stop writing here, so I'll stop writing there, okay? Yes, exactly, Tanuja. That's my target for today. Okay, when I started the class, that was my target. Yes, tell me. Ready? Yeah, PYQs will solve, Siddha. That's coming. Don't worry. Okay, pistil or carpel? Female reproductive part we are talking about. So, what is our female reproductive part? Map, it is gynoecium. Gynoecium is made up of units of carpels. Yes or no? Units of carpels. Now this carpels have three parts. What are the three parts? They are going to have a landing portion where the pollen grains are going to land. They are going to have a long stalk like structure that is going to connect the ovary and the stigma, right? This is going to be my carpel. This is going to be my carpel. Okay, doubt ma'am. Can we grow plants with tears instead of waters? Okay, yes. Like how you are growing without brain cells, you can definitely grow plants with tears. Okay, yes. This is to one of my very favorite student in the class, students. So do not be like, what did ma'am tell? Brainer. Okay, do not think the student is like this. Okay, yes, very nice doubts. He has doubts from space. Okay. Let's do experiment. Before May 5th, you better do the experiment and finish. Okay. After May 5th, I will come there and I'll hit you people and make you cry. So listen to the class now. Okay. All space species there. Okay, come, continue. So this is the stigma, this is the style, and this is the ovary. Okay, it might look like a flower vase, but please understand this is the carpel. Okay, what is the first portion called as? It's called as stigma. Okay, it's called stigma. This is the where the pollen grains are going to land. Okay, pollen grains land. Okay, this is the landing place for the pollen grains. This is called the style. The style, you can students call it as a connection. Okay, this is the connecting link. Okay, this is a connection or connecting link from stigma to ovary. Okay, stigma to ovary. Got it? Yes or no? Clear? Is this clear? Okay, that also I'll lend you people after some time. Okay, this is ovary. This is ovary where the ovules are going to be present. Okay. Any doubts so far? This is perfectly clear? Yes or no? Now students, this portion is called as the locule. This ovarian, or we can call it as ovarian cavity also. So, ovarian cavity is where, or your locule is where your ovules are going to be present. Okay, it is going to be present in the placenta. So, this portion will have the placenta, and inside this your ovule is going to be there. Okay, inside this your ovule is going to be there. Clear? Yes or no? Plants will get mineral deficiency, it seems. People are serious about crying and growing the plant. Huh? Okay, is this clear or not? This is your carpel. Now students, your carpels can be attached. Okay, they can be single. Okay, like you people, they can be single. So that is going to be carpel. It's going to be monocarpellary. Okay, monocarpellary. If they are fused, okay, that means if they are many in number, they can be fused or free. Okay, they can be fused or free. So when they are many, they are called as multicarpellary. Okay, multicarpellary. And if they are multicarpellary, right, if they are multicarpellary, they can be fused or free. Fused or free. Free will be fused, students. What is it called? Syncarpus. Okay, fused is called syncarpus and free is called apocarpus. Apocarpus. How did I tell you? You can remember the example. How did I tell you? You can remember the example. I told you, you can remember like this. Syncarpus, apocarpus. You can remember MSP and MAM. Ma'am. Right? MSP and MAM. Ma'am. Okay. MSP means multicarpellary syncarpus papaver. Papaver somniferum is the example. Okay. MAM means multicarpellary apocarpus michelia. Okay. Michelia. Is this clear? Yes or no? So, the examples also is here. learn only this much, you are done. Okay, you are done. Okay, this is going to be the ovule, and this portion where this ovule is going to be attached, no, this portion we call it the placenta. I think this and all you have studied in your morphology. Okay, placenta. Clear? Okay, this is where your pollen grains will come and fall. Landing. Done. Okay. Okay. Chalo, students, now look here, this is the examples as I told you. Okay, this is going to be your how stigma style ovary is going to be there. This is going to be apocarpus, this is syncarpus, this is going to be apocarpus, and here is your ovary. Ovary we will draw now, and here examples are given. See, even though it is given in the picture, they will ask you questions. Okay, so please remember MSP and MAM, ma'am, and MSP, very, very important. Okay, shall we draw ovule? Now ovule is a task, students, for me to draw. Okay. Ovule is a task. Now I will do jugaad. Yes. Does it look like an ovule? Yeah, it looks like an ovule, no? So this is for understanding. Okay. This is your embryo sac. This is the most important one. Fine. Oof. Sorry, students. 1 second. This integuments are wrong. If integuments are wrong, how can I'll draw a smaller one then. Okay, this is better. Okay, yes, we got a better overview. Okay, yes, Navratri series day 4, that should be your comment. Okay, Navratri series day 4 completed. Okay, done and dusted. Okay, that's what your comment should be for today. If you do not comment, then I have nothing to say. Okay. Okay, chalo, let's start labeling this. This is an embryo sac. Now what is, sorry, this is an ovule. Okay, inside that embryo sac is there. Why am I calling this an anatropous ovule? Okay, why am I calling this an anatropous ovule? Because, okay, because anatropous ovule is because when the funicle, micropylar end and chalazal end is not in a straight line, we call it as anatropous ovule. Okay, so if you see your micropylar end and chalazal end is in a straight line, but funicle has gone that side, so that's why we call it an anatropous ovule. Now students, let's start labeling. All of these person has a job in the next. Okay. Okay, now students, remember, all this has a job in the future, so we have to know each of them. Okay, we have to know each of them. So as I told you, this is the funicle, which is like a stalk. Okay, which is like a stalk that is going to hold your entire ovule. Okay, it's like a stalk. Then these are your two integuments. Okay, this is going to be your outer integument, and this is going to be your inner integument. What are they going to do, students? These people are going to do the job of protection. These people are going to do the job of protection. And later they will form tegmen and testa. Okay, testa and tegmen, that is the seed coats will be formed because of these two people. Now when I go inside, okay, before that there is going to be a layer. Okay, students, you can draw this way or you can draw this way also. Okay, you can draw this way also, no problem. If I hold it like this or if I hold it like this, is the same only. So I am not drawing this way. Many people will teach you this way also. Both are right. Okay, both are right. Okay, I can either draw it this way or upside down. Now students, remember that where the funicle is attaching, okay, attaching to the entire ovule, it is going to be called as your hilum. Okay, so this portion, okay, this portion where the attachment is going to happen, that is called the hilum. Hilum. And this hilum later in the seed, have you seen in the seed, students, where the middle portion, this is a funicle, the stalk, okay, the middle portion where it is going to absorb the oxygen and water is going to happen because of the hilum. The seed also, there is a small pore in the center, if you have noticed in your rajma and all, if you have noticed, this is how your kidney beans is, no, if you have noticed here, you will see a small circle, okay, and then you will see a connection. This is actually a hilum, a small pore through which the oxygen and the water will enter inside the seed, okay, and allow them for germination. Got that? Yes or no? Okay, clear? Sahil, it will take 2 decades for that to happen. Okay, chalo. Next one, hilum is done. Funicle, outer integument, inner integument. Students, the diagram might not be perfect, but labeling is very, very important. Now, in the middle, you have the embryo sac. Now, this embryo sac, remember, this embryo sac has a covering, okay, of cells. So, let's say if I draw one more layer like this, okay, one more layer like this, and I am going to draw a set of cells here. These cells are called nucellus. What are they called? Nucellus. And this is going to provide the nourishment for the growing embryo sac. Okay, this is going to provide the nourishment for the growing embryo sac. Got it? This is providing the nourishment for growing embryo sac. Clear? Yes? Okay. Now, students, inside this embryo sac, if it is not mature, then we are going to have only one cell, that is called as your megaspore mother cell, where one cell will become the megaspore mother cell. Otherwise, this embryo sac is going to differentiate, and they are going to have 3 antipodal cells, okay, 2 synergids, okay, 1 egg cell, okay, 1 egg cell, this together forming the egg apparatus, okay, and middle they are going to have a 2 polar nuclei. This is how a fully developed embryo sac is going to look like. Okay, fully developed embryo sac is going to look like. Very good. Female gametophyte is your embryo sac, because inside the embryo sac is what the egg is going to be there. Yes or no? So, this is going to be your embryo sac, okay, which is going to be your female gametophyte. So, students, in Megasporogenesis, okay, in Megasporogenesis, there is going to be more mitotic divisions, okay, where there will be 1 meiotic divisions and 3 mitotic divisions. In male Microsporogenesis, you saw 1 meiotic division and 2 mitotic divisions, right? So, always remember, okay, try to connect. So, this is the embryo sac where there is a female gametophyte, okay, female gametophyte. Funicle is just a stalk, dear. Okay, funicle is just a stalk. After that, they will disappear. Okay, they will start degenerating and they will not be required. No, but other people will have a job. Other people will have a job. Now, what I wanted to write? Yes, students, inside this, okay, remember that one cell. Okay, one cell is going to become the MMC, megaspore mother cell. That megaspore mother cell is going to form the megaspore. Okay, megaspore, and that is going to form the egg and embryo sac. All the divisions is going to happen. Clear? And anatropous ovule is clear. So, this is what we just learned. See, here it is given this way. I just twisted this. Okay. So, here you can see embryo sac, inner integument, outer integument, micropylar end, and wait, I have to write that, no, micropylar end and chalazal end. So, students, this is going to be the chalazal end. Chalazal end. Okay. This is going to be the micropylar end. How do I know it is a micropylar end? Because there is an opening for the pollen tube to arrive. Yes or no? Where the integuments are absent, that is going to be your micropylar end. Always you are going to identify with the help of that. Okay, where the micropylar end is going to be, that is where the pollen tube is going to arise. Yes or no? Clear? Yes, we will go further. Now, Megasporogenesis. Ready for Megasporogenesis? So far good, right? Yes, any doubts? All of you tell me in the chats faster. Okay, pakka. Chalo, clear. Okay, ma'am. Okay, yes. Now, students, Megasporogenesis. So, ovule is going to be there. I told you, inside that ovule, one cell is going to become potentially the megaspore mother cell. Okay, it's going to become the megaspore mother cell. Okay, this megaspore mother cell is going to undergo meiosis. Okay, is going to undergo meiosis and form megaspore tetrads. Okay, you'll be like, what, ma'am? These people also form tetrads. Yes, students, these people also form tetrads. That means 4 of them will be formed. But unfortunately, out of the 4, only one becomes functional. Only one becomes functional. Okay, only one functional megaspore will be there. Is this clear? Okay, only one functional megaspore is going to be there. 3 will degenerate. Okay, 3 will degenerate. So here I'll write, I'll do one thing. If I erase this and this arrow mark. Yes. Yeah. So out of this, I'm going to write this way. 1 becomes functional. Okay, 1 becomes functional and 3 of them will degenerate. Okay, 3 will degenerate. Okay, yes. Now this is going to undergo mitotic divisions and this one functional is going to form your female gametophyte. Okay, embryo sac. From embryo sac it is going to continue. Any doubts so far? No, right? All perfectly fine. So this is going to become into the first step, that is going to be 2 nucleated stage. Okay, 2 nucleated stage. From 2 nucleated stage, students, it's going to become 4, 4 to 6, 4 to 6 to, sorry, 2 to 4, 4 to 8. Okay, 2 nucleated stage, from 2 nucleated stage to 4 cell stage, 4 to 8, and then it is going to form your embryo sac. So from this 2, it is going to form 8 nucleated, okay, 8 nucleated 7 cell stage. That is your embryo sac. Perfect, perfect. So let's write here first meiotic division. Okay, first meiotic division. From here, students, after the megaspore mother cell functional happened, first mitotic division, and here again 2 mitotic divisions are going to happen. So 1 meiosis and 1 meiosis and 3 mitosis. Okay, 3 mitosis. Clear or not? Eight nucleated cell stage will be formed. That 8 nucleated stage will have what and all? Your antipodals. Okay, it will have antipodals. 3 antipodals will be there. Okay, 2 polar nuclei will be there. Students, Josh is very less. Okay, I am losing my energy because you people don't have energy. 2 synergists are going to be there, plus 1 egg. Okay, plus 1 egg. So this fellow will technically be the embryo sac. Got it? Done? Ammu ma'am is substitute. Such nonsense these people are telling. Don't believe them. Okay, I am only Gopika ma'am, I am only Ammu ma'am, I am only attendance ma'am, I am only Mintu ma'am. Okay, all these names have been given by my students. Yes. So students, up to here, up to the formation of megaspore tetrad is going to be a megasporogenesis. Okay, after that it is not going to be called megasporogenesis, right? So up to this is going to be a definition for megasporogenesis. Sporogenesis. Clear? Yes? Okay. Chalo. Okay, this is what has happened. Ovule, 1 cell became megaspore mother cell. After meiotic division became megaspore tetrad. 1 became functional, 3 degenerated. That functional one went through mitotic division from 2 cell stage to 4, 4 to 8 cell stage through meiotic divisions itself. Okay, so free cell stage they are going to form. Now embryo sac, students, embryo sac we just drew the same structure we are going to draw here, but an enlarged structure to make you understand. Okay, an enlarged structure to make you understand. See, this is your embryo sac. Okay, in this embryo sac, students, remember the stages I will show you, the diagrams I am going to show you. Okay, diagrams I am going to show you, but what was there? See, 8 cell stage was there. Okay, let's just draw one more cell here. Okay, this was the before cell. Okay, before cell, this cell went to this cell. Okay, so in this stage what was there? 4 cells here, 4 cells here. This was an 8 cell stage. Okay, this was an 8 cell stage. One second, students. Okay, this was the stage, 8 cell stage. Now from this 8 cell stage, they are going to separate. They are like, okay, you know what, this is not going to work, so we are going to become properly, we are going to arrange ourselves properly. Okay, we are going to arrange ourselves properly. And for that, okay, for that, 3 of them went to one pole, which is called as the antipodal cells. Okay, 3 cells went to one pole, which is called the antipodal cell, and that end is called the chalazal end. Okay, then 3 of them came here also. They called themselves the synergids. Okay, they were the synergids, and in between came our heroine, that is our egg apparatus. Okay, like the Pooh and the friends, no students, in the movie, I forgot the movie name only, Kabhi Khushi Kabhi Hum. Okay, whatever. Okay, like the Pooh, that Karina has 2 friends, no, like that, these people, synergids plus the egg. Then 2 cells, okay, 2 cells of these, okay, these 2 came together, okay, came together to form the polar nuclei. Is it clear to all of you how embryo sac is formed? Okay, polar nuclei. Now in the synergids there is a specialized structure, okay, in the synergids there is a specialized structure called filiform apparatus, who are like the people who are telling pollen tube, please come this way. Okay, please come this way. Are you all listening? Please come this way. This is where you have to go. This is where you have to do the fusion. So here they are going to do that. So let's label. This is going to be your antipodal cells. Okay, this is going to be your antipodal cells. And which side are they present? Which end are they present? They are present on the chalazal end. Okay, chalazal end. Then this side you have the micropylar end. Okay, you have the micropylar end. Now students, this 3 became the antipodals, this 2, okay, became the polar nuclei. Polar nuclei. Okay, and this, there is a central cell. Okay, one more cell is there like this, which is called the central cell. Okay, I am just labeling it here only, central cell. And this is going to be your egg. This is going to be synergids. Okay, synergids, and they are together called, okay, what are they together called? Egg apparatus. Egg apparatus. Yes, perfect. Yes or no? Yes ma'am, listening. Okay, all of you are not listening, only some of you are listening. Okay, I can understand. I can understand everything, I can understand. Chalo. So this is chalazal and micropylar end. This is how your embryo sac is going to be there, and this is going to be a filiform apparatus. Okay, this is going to be filiform apparatus, who are the guiding light. Okay, guiding light, like you will have a relative, no matter whatever exams happens in the world, they will get to know if you are writing or not, and they will call you first. Eshtiде marks chenagidiya? Kitna hai marks? Aise they'll ask, no, in all the languages. Okay, epidemic marks. So like that, different language, they'll ask you, how was your marks? You know what, my son got 2 marks extra than you. Same way, this filiform apparatus is the person who is like, come inside, pollen tube, you come this way. So they are like the torch bearers for the pollen grains to enter. Any doubts, students? Any doubts in this? Tell me. So this is your Megasporangium from your NCERT, okay, NCERT. So here you can see, in a typical angiosperm ovule, the ovule is a structure attached to the placenta by a means of a stalk called as funicle. As I told you, it is a stalk, okay, stalk called as funicle. The body of the ovule fuses with the funicle in a region called as hilum. So body of the ovule, that ovule and funicle is attaching in a portion called as hilum. Okay, yes. Thus, hilum represents the junction between the ovule and funicle. Each ovule has 2 segments, that is the outer integument, inner integument, both are going to do the job of protection. Okay, both are going to do the job of protection, and they will have a small opening which will lead to the micropylar end so that the pollen tube can enter inside. Now, enclosed within the integuments is a mass of cells which is called the nucellus. This nucellus is going to provide abundant reserve food materials for the growing embryo. Okay, growing embryo. Clear? Now, the embryo sac or the female gametophyte, and ovule generally has a single embryo sac formed from a megaspore. One functional megaspore form that one embryo sac. Okay. Now, megasporogenesis, students, the process of formation of megaspores, okay, from megaspore mother cell, okay, from megaspore mother cell is called as megasporogenesis. Megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region. So, look here, this is the division I told you, I will show you. See, here you can see, nucellus is there, this is the one who is going to provide the nourishment. This is your megaspore mother cell. That megaspore mother cell form the dyad, then form the tetrad, but dyad is not mentioned anywhere. So, directly you can go for tetrad, okay, directly you can go for tetrad, megaspore, tetrad. Now, students, after that what has happened? See, here is where your mitotic divisions are happening, okay. So, 1 meiotic division, 3 mitotic division, there 1 meiotic division, 2 mitotic division. So, some student asked me, no ma'am, how many microsporogenesis, megasporogenesis, how many mitotic, meiotic division? Listen to me carefully. So, students, there one happened, that means from the microspore tetrad, one became functional, no? That one functional became 2 nucleate, that became the first mitotic division. Then this is the first mitotic division, this became the second mitotic division, this becomes the third mitotic division. So, after 3 mitotic divisions, okay, after 3 mitotic divisions and 1 meiotic divisions, we get our embryo sac. Okay, we get our embryo sac. This is our embryo sac. They will arrange themselves, they will move to the poles and they are going to be like, okay, this is my arrangement, I am going to work according to this. Okay, I am going to work according to this. Okay, first question, all of you answer me one question. Let us see how many of you understood, and then we will go to pollination. Okay, we have formed the gametes, students, now we have to make them fuse together. Okay, we have so much again, let us do it fast. Answer the questions fast, then we can speed up. Okay, I am going to wear my glasses, it has been some time that I am not. Where have we reached in our targets? Okay, what's the answer? Which of the following layers in the walls of the anther nourishes the developing pollen grains? Very good, very good. Answer is going to be tapetum. Okay, next question. A typical angiosperm anther is dithecous, which means that it is dithecous, which means what? Yes, Sahil, let's do that. Tell me, a typical angiosperm anther is dithecous, which means that it is GA. I'll hit you people. 2-lobed and each lobe has 2 theca. 2-lobed, each lobe has 1 theca. 4-lobed and each lobe has 2 theca. A very easy question, but if you do not think, you can go wrong. You can go wrong, 100% you can go wrong. What is the answer? Yes, answer is 2 lobes, right? A typical angiosperm is dithecous, that means they have 2 lobes and each lobe has 2 thecas. Very good. Okay, very good. Last question for now. Pollen intine is made up of? Pollen intine is made up of? Faster, yeah, easy question it is. Very good. Very good, very good. How many of you got full marks? Easy Pepsi it seems. Okay. And told the wrong answer. Now students, we have coming to the last step of pre-fertilization, that is called gamete transfer, which is going to happen with the help of pollination. Yes or no? What is pollination? That means transfer of pollen grains. Transfer of pollen grains. From where to where? From anther to stigma. It could be of the same plant or it could be of a different plant. So, if it is going to be with the same plant, okay, that is same plant, I will call it self-pollination. I will call it self-pollination. Yes or no? All this you have studied in your smaller classes. And if it is going to be from to a different plant, I will call it cross-pollination. So, here what is given in your textbook? They have used the word autogamy and geitonogamy, xenogamy, right? Xenogamy. Correct? Okay. So, same plant self-pollination, different plant cross-pollination. Correct or wrong, students? I'm correct, right? They'll be like, hey ma'am, you're correct only, obviously. Okay, yes, fine. Okay, so same plant. So here students, they have given two types of pollination, which is called as autogamy. Students, auto means what? Auto means three-wheel vehicle. No. Auto mode means what? You take the phone, you keep it in auto mode, everything light, everything contrast, brightness, everything will be adjusted by the phone itself. That is called as auto mode. That means by self. So autogamy also means self-pollination. That is from the anther, okay, anther of the same plant, it is falling on the stigma of the same flower, okay, stigma of the same flower. So, this is called as self-pollination. This is called as self-pollination. Now, what is called as geitonogamy? That is, they have used the word geitonogamy. Geitonogamy is nothing but, imagine this is a plant, okay, this is a plant, there is a flower here. Such a bad combination of flowers, students. Okay. Okay, this is a flower. One flower is here. Okay. One flower is here. Okay. And let's say one more flower is here. Okay, these are the flowers. Now, let's say this is male. Okay. And this is female flower. Now, what is going to happen, students? From the anther of this flower, the pollen grains will fall on this flower. That means in the same plant, the pollination is happening between two different flowers. That is called as geitonogamy. Last one is xenogamy. Xenogamy means what? So, I will just connect this, okay? Otherwise, this image will not make any sense. Okay, this is the same plant. Now, imagine xenogamy, okay, xenogamy, cross-pollination other word, okay, cross-pollination other word. That is, this is going to be the stigma and the anther from different place because of an insect or something, the pollen grains have come and landed on this. Clear or not? Is this clear? Yes. Okay. Yes. So, this is just different flowers, same plant. This is different plant by itself. Okay. That's why I have written only cross pollination and self pollination. I have not written anything else. Okay. Is this clear? Auto, you people drive that. Okay. Pollination is clear. Now, kinds of pollination. That's what I told you. So, let's just delete this. Okay. Kinds of pollination also I told you, 3 types they have given. I think 3 types also you understood, no? Here self-pollination, autogamy, geitonogamy, different flowers, xenogamy, totally different. So, here we have the given to us. Okay, so students, one second, here 2 more things I have to give you, no? What are the 2 examples that they have given you? What is happening here? What are you people talking about? I am teaching, you're talking. Good students. Okay, ma'am is teaching, you should talk only, no, obviously. If ma'am is teaching, what else will I do, ma'am? Yes. So here students, they have given you an example of under autogamy. They have given chasmogamous. Yes or no? Chasmogamous, and they have given clistogamous. In this chasmogamous flower will have an exposed anther. Okay, it will have an exposed anther. They can be open or closed type, students. If they are opening also, they will not favor cross pollination, they will go for self pollination. Okay, self pollination. Yes. Now here clistogamous means they will never open. Okay, these flowers never open, because of which they have no chances of cross pollination. They will always go for self pollination. Imagine a flower does not open, how will the pollen grain come and fall? The butterfly will be like, can you please open, please, I have pollen grains, can I dust it? They are not going to talk like that. No, they are not going to have any interaction. So since this flower is never going to open, this flower is going to be closed. So what is going to happen? Only self pollination is going to happen. Is this clear to all of you? Yes or no? Yes or no? No or yes? How come they haven't given examples here for you people? Okay. Why is Selvi talking about food here? Okay, why are you talking about food? Students, these 3 examples they have given, no? Commelina, oxalis and voila. Right, these 3 examples only is given, right? Yes, voila, oxalis and commelina. Okay, this is the examples given. Ready? Okay. So students, because these people are doing self pollination, variations are not going to be there. Okay, think about this like COVID. Okay, COVID you remember, in that I and D you forget. Okay, I and D you forget, and you remember only cove. So these 2 students, what is going to happen? No variations are going to happen. No variations are going to happen because these people are not ready for cross pollination. Only cross pollination, just like asexual reproduction and sexual reproduction, autogamy and xenogamy has their own advantages and disadvantages. Autogamy will not lead to variation because it's the same thing. It's like marrying inside the family and telling that, hey, you know what, that mama's disease I will not get because you know what, I am mama's ponnu, so I will not get the disease. Does not matter, you are from the same family and that genetic disorder can come to you or it can come to your kids. Okay, so just by telling examples or telling that how far you are related to that person will not escape. Same thing will go here. Just because telling that a different flower I am on the same plant, I am a different flower, I will have variation. No, you are on the same plant itself. Okay, yes, clear? Is this clear to all of you? Chalo. Now students, ayo, not chalo. Now who can pollinate, right? Pollinating agents. Pollinating agents can be 2 types, abiotic and biotic. Okay, biotic. Now abiotic, okay, under abiotic you have wind and water. Out of that, wind is the most common. Okay, water. Under biotic you have insects, flies. Okay, insects are there, students. Under insects itself you will have flies, beetles and all of that. Now, and even humans are potential, pollinators. Like we eat a fruit, we throw the seed, that is different. But here what we do, we sometimes with our dress itself we take the pollen grains and go. Okay, yes, got it? Clear. Now this one, okay, what did I tell you? Wind pollinated flowers can be called. I gave you some words, entomophily, hydrophily. Any of you remember? Who's going to tell me the answer? Quickly in the chats. Let's see who is remembering. I'll give you a clue if you want. A. This starts with A. This starts with H. This starts with E. These 3 names only you have to remember. Theriyum. Tamil theriyum. Okay. Anemophily. From wind you call that as anemophily, hydrophily and entomophily. Okay, entomophily. Clear? These 3 are the most common pollinating agents. Now, according to NCERT also, you only have to study these 3 agents. Now, I am going to mark the words that is important for you. Only those words you have to focus. Okay, clear? Now, pollination by wind is more common amongst the abiotic pollination. That means compared to water, wind is the most common. Okay, wind is the most common. Wind pollinated also requires the pollen grains. So, first characteristic of a wind pollinated plant is that the pollen grains have to be lightweight, otherwise you cannot carry this fellow and go somewhere, right? Okay, and they should be non-sticky, because if they are sticky also, wind cannot take them and go. They'll be like, hey, I'm not coming, you take me, I'll not come. It shouldn't be sticky, it should be lightweight and should be non-sticky. So, as soon as the wind comes, it will take and go. Now, so that they can be transported in the wind currents. They often possess, now, the plant which is supposed to take the pollen and have the pollen, they both should have an exposed stigma, should be a little more height so that the pollen grains can land easily. All this adjustment the plant has to do. It should have a well exposed stamen, okay, so that it will be easy for the dispersal, and they should have large, often feathery stigma. Okay, that's why? So, pollen grains can be easily trapped. Okay, pollen grains can be easily trapped. To easily trap the airborne. Now, wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. Because here beauty does not matter, brain matters, because wind has to take it, not insect. For insect, we need to put colorful petals. Okay, a familiar example is of the corn cob. So, the tassels you see here, students, there is an error here. The tassels you see here are not, see, the tassels you see here are nothing but the stigma and style which wave in the wind of the trap pollen grains. This is actually a little error is there. I'll show you the diagram. I'll send the image in the telegram and I'll tell you what it is. Without showing the image, I will not be able to tell you what is the problem. The tassels are there and the stigma is present near the ear of the plant. Okay, so it's a small change, error here. So, I'll let you know about this. Okay, so wind pollination is quite common in grasses. So, you can remember this, grasses, please remember. In monocot, especially in grasses, wind pollination is very important. Students, only these words you have to focus, nothing else. Whatever I have underlined, stick to these words. Okay, these are the features of your wind pollinated. Now, water. Okay, hydrophily. Students, only 30 genera, okay, of plants are going to do water pollination, because it's very rare. Now, as you saw in bryophytes, to complete their one full life cycle, they need water. That is why we call them amphibians of the plant kingdom, right? Why do we call them amphibians of the plant kingdom? Because this fellow needs both land and water. Water he needs to complete its one full life cycle. So, same way, very few genera are there which is going to be, pollinate in water, because of the fact that it is not very easy to pollinate in water. Water currents will take the pollen grains and go. Got it? Chalo. Now, as against this, you would recall that water is regular mode of transport of male gametes among the lower plants like your bryophytes and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transport of male gametes and fertilization. So, it's a fact that stopping them from spreading so wide or becoming more developed, because these people need water and it is kind of like a barrier. If you need water and you're like, no, without this I will not do this, without this I will not do this, that means what, you're too specific and you're too picky, right? You have a best friend, you're telling her, you know what guys, I don't want to talk to her because you know she sneezes loudly, she laughs loudly, she looks at me like this, she has a brown hair. If you be picky like this, no one is going to be friends with you. Same thing goes here. Since bryophyte and pteridophytes always require water for completing their life cycle, they are not able to spread as much as, like, understand. And you understood, no? Yeah, obviously you will understand. Now, some examples of water pollinated plants are your Vallisneria and Hydrilla. Hydrilla, you remember in photosynthesis of plants, lot of experiments we are doing with the help of Hydrilla. Now, which grow in freshwater and several marine seagrasses such as Zostera. This is the only seagrass example given here. So just remember these 3 examples. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth and water lily, the flowers emerge. So in water hyacinth and water lily, if you would have seen, students, you would have seen that purple color flower. Okay, water hyacinth flower is purple color. That fellow will come up. Okay, he'll come above the water and he'll be like, you know what, I'm ready for pollination. Please come and pollinate me. I do not want to do it down. So they have bright colored flowers. Okay, you just notice it when you're going to your village and all. The flowers emerge above the water and are pollinated by insects or wind. Okay, so not everyone who is staying in water is dependent on water. Only 30 genera. That question can come, so be careful. In Vallisneria, the female flower reaches the surface of the water by a long stalk, and the male flowers or the pollen grains, see, the pollen grains are released on the surface of water, and remember they will have a covering, okay, so that this does not get wet. Pollen grains shouldn't get wet, no? So they will have a covering, a mucilaginous covering to help them not to get completely wet by water. They are carried passively by the water currents. So in Vallisneria, female flowers will come up, okay. How are they going to come up? With the help of a long stalk, okay. They will reach the surface. Please learn what I have underlined, okay. Long stalk, and the male flower pollen grains will be going in the water, and because of the water current, they will reach the female flower. Some of them eventually reach the female flower and the stigma. In other group of water pollinated plants like sea grasses, like your Zostera, what is going to happen? Students, that got cut, yaar. And that snip went off. And let me just check, it got mixed up or somewhere. No, I guess. 1 second, just give me 1 minute. That got cut. Let me just open NCERT 1 minute. I don't know what happened. The slide was there. Yes, just give me 1 second. Okay. Okay. Okay, that entire ribbon like portion got cut. Okay, I was just checking what got cut. Okay, what got cut. So what is happening, students? Here in sea grasses or some of the plants, what is going to happen? The pollen grains will be released in the water itself. In these kind of ones also, there is going to be mucilaginous coating so that they do not get wet and they will float by the water currents. But the chances of them reaching to the female flower is very rare, okay? Because they are going in the water current, anything can eat them, they can get stuck in anything. Corals will be there, that will be there. If it is freshwater, plants will be there, you know, all the zooplanktons, all of them will be there. The roots, to their roots, they can get attached. So the chances of them getting fertilization or pollination to happen is very rare. That's why most of the plants will go for insect or wind pollinated, because the chances of reaching will be for sure, okay. But in water, that's not the case. So here they have given about the mucilaginous covering, that last paragraph, that is what I was just checking, okay. So that is all there for you. Now students, insect pollinated, this is one of the most common biotic factors, insect pollination. Majority of the flowering plants use a range of animals as pollinating agents like bees, butterflies, flies, beetles, wasps, moths, birds, hummingbirds, etc. Bats are also common pollinating agents, okay. Bats are also common pollinating agents. Now, among animals, insects, particularly the bees, all of you know honeybees. Okay, they get nectar as the reward for pollinating. They get the nectar telling that, hey, thank you so much, you have given me the nectar, I have pollinated for you, and they will make honey out of it, right? So, are the dominant biotic pollinating agents. Biotic pollinating agents. Even larger animals such as you have primates, okay, arboreal, tree climbing ones, rodents, all of this also does pollination, okay. This question has come, students. Rodents and your lizard, garden lizard, do they do pollination? Students did not know this, so just keep in mind they are also reported as pollinators. Okay, they are also reported as pollinators. What are you people talking? Now see, majority of this side I have to stand. Okay, majority of the insect pollinated flowers are large. Okay, imagine here, these are the main points. Okay, they are large, colorful, fragrant, okay, rich in nectar. These are the most important factors for the insect to come. If you do not give nectar, insect is not going to come. That's a reward. Okay, that's a reward for doing pollination, because if pollination does not happen, this whole chapter we are studying about the importance of sexual reproduction becomes a question mark. Okay, so when the flowers are small and the number of flowers are clustered into an inflorescence to make them conspicuous, that means to make the insect attract. Okay, animals are attracted to the flowers by color. Okay, not like our some of the class students who are telling red and putting green hearts, telling green and putting red hearts. Okay, they are correctly attracted to colors. Okay, yes, the flowers pollinated by flies and beetles. Students, flies and beetles, as you always know, these people are actually carriers. Especially your flies are carriers of disease. These people are not going behind colors or anything. So they will go for a foul odor to attract these animals. Okay, they will secrete foul odor. Now if you see a rat is dead, a crow is dead on the road, what do you see the most common around them? Flies around them. That means what? Flies are attracted to odor. Okay, to bad odor. So the plants will be like, I need to get pollinated from beetles and flies, so I am going to secrete an odor. Okay, which is a bad one. Got it? So to sustain animal visits, the flowers have, very important question, flowers have to provide rewards to the animals. And what are the rewards? Nectar and pollen grains are the rewards. Okay, flowers have to provide, okay, rewards. What are the rewards, students? Pollen grains and nectar. Okay, pollen grains and nectar. Wherever possible, I've added your NCERT snip, okay, because writing all this will take one eternity, and no need to write all this also, you just have to know the words of NCERT. Okay, so maximum places I've used NCERT snips where we can go through NCERT and I'll make you understand. Nectar and pollen grains are the usual rewards. Very important, nectar and pollen grains are usual floral rewards. For harvesting the rewards from the flowers, the animal visitors come in contact with the anthers and the stigma. The body of the animals get the coating of the pollen grains. So like how we have Sony bee in the class, like that honey bee will be there. Okay, this honey bee's body, right, this is the body of the honey bee. This body of the honey bee is going to have fur, okay, like a small substance. To this, what is going to happen, students? Your pollen grains are going to get attracted. Okay, this pollen grains are going to sit on this, and they will not know, knowingly, unknowingly, they will dust this pollen grains on the stigma of another plant. Okay, they will dust this pollen grains on the stigma, and if they are compactable, then they are going to continue the fertilization. Okay, this pollen grains will come and land. Is this clear? Okay, now some mutualism, commensalism you are going to study. In some species, okay, floral rewards are in providing, very important, in some species floral rewards are in providing safe spaces to lay eggs. Yes or no? They are telling that I do not have anything to do with your nectar or pollen grains. You know what? I want to lay my eggs. Let's say a moth is talking to a plant, and the moth is telling, you know what, if you can give me the space in your ovary, I will lay eggs there, and for that I can take the pollen grains and go. I can carry the pollen grains, or in other way around, some kind of mutualism or some kind of help for each other. Okay, so in some species floral rewards are in providing safe spaces for animal. Example, the tallest flower called as Amorphophallus, the flower that is around 6 feet, okay, taller than all of us. A similar relationship exists between them, species of a moth and a plant, yucca. Okay, Pronuba moth and yucca plant also, they have the same thing, where the moth will lay the eggs and yucca plant is going to protect the eggs. Yucca plant is going to protect the eggs. So moth and the plant cannot complete their life cycle without each other. The moth deposits its egg, where, students? In the ovarian cavity, which the plant should not, like, it does not provide, that is for the ovules. But what they will do, they will help in providing that, giving that ovarian cavity, telling that you lay the eggs here. Okay, I will protect it. That is the floral reward they are getting. So these people have become so dependent that without each other they cannot complete their life cycle. So who is the example here, students? It is your yucca, okay, yucca plant plus your Pronuba moth. Okay, these people need to go through their life cycles. Okay, so what is going to happen? The moth is going to deposit its egg in the locule of the ovary, and the flower, and in turn gets pollinated by the moth. So, the flower will in turn gets pollinated by the moth. Clear? Yes or no? So, what are the floral rewards? Pollen grains, okay? Pollen grains. Okay, so it's going to be pollen grains, nectar, and also floral rewards could be where they're telling that I will take care of your ovule, or I'll take care of the eggs, you please pollinate and give it. Okay, pollinate and give it. Okay, thank you so much. So, all the floral rewards are clear. No, this is very, very important. Okay, very, very important. Thank you, Rasna. Nectar, pollen grains, and safe place for laying eggs. Okay, done? Yes? Students, I did not get water. Will you wait for 1 minute? Can I get some water and come? 1 minute, okay? Thank you so much, Madhumati and Rasna. People are making me emotional. Yes. Juxtaglomerulus to work in out seems. Okay, ready? Clear? Can I continue? Okay. Another topic, students. Okay, another topic. This chapter is huge chapter, no? That's why so many topics are there, but all are very interesting topics. Yes or no? Okay, let's start. Okay. Now, students, listen here. Outbreeding devices. Before you knowing outbreeding devices, you have to know something called as inbreeding depression. What you have to know? Inbreeding depression. Inbreeding depression means what? A plant can go through inbreeding depression when the plant goes through a continuous, okay, continuous self-pollination. Continuous what? Self-pollination. So, students, when the plant goes through continuous self-pollination because of no variation, because of lack of variation, the plant will feel frustrated. Okay, the plant will be like, oh my god, no variation, same faces again and again I am seeing. So, the plant will be like, I am tired, okay, I am tired. So, that is called as inbreeding depression. To come out of inbreeding depression, the plant is very smart also. Plant is not going to be like, oh, do I have depression or I have to go out. Okay, plant will not rethink like that. Instead, plant is going to do one thing. Plant is going to find some outbreeding devices. Okay, outbreeding devices. Yes, due to continuous self-pollination, plant is going to go through depression, which is called as inbreeding depression. To get out of inbreeding depressions, okay, students, I learned this arrow mark, it's very nice, no? To come out of inbreeding depression, they go through outbreeding devices. Outbreeding devices means devices that are going to help the plant to undergo cross-pollination, okay? Help plant undergo cross-pollination, okay? Cross-pollination, okay? I help plant undergo cross-pollination. Is this clear? Okay, is this clear? So, let's start with one of them, okay? Who is feeling sleepy in the class? Stand up in the class, raise your hands. I saw, okay? Before the message got deleted, I saw. Very smart. I'm like a maths teacher in the school that you didn't want, okay? I love to be a strict teacher, you know, scolding me this. But the thing is, I cannot control my laugh. So if I scold after 2 minutes, I'll laugh. So it has become wasted. Okay. So that's why I just left the thought of being a strict teacher. So now I'll be strict with all of you. Now listen to me carefully or I'll scold you people. You listen or not? Now they'll be like, ma'am, we won't listen. Students, I am also simultaneously checking this also, okay, the cards. So I can tell if there is any correction required. Okay, when I am teaching, I am just going through the cards also to see if all the contents are there. So some contents are there. Okay, yeah, chalo. Okay, to get out of inbreeding depression, first thing the planters do is very smart move, students. Very smart. Even you wouldn't have thought of this, but the plant is like that. First thing the plant will do is, to avoid self-pollination, it is going to change the different positions, okay. It is going to make different positions of anther and, okay, positions of anther and stigma. See, all of these conditions have names, but you do not have to know, okay. All these conditions have one name, which you do not have to know. So what they are going to do? Before situation was like this, okay. Before situation was like this. Yes or no? Like this the situation was, so the pollen grain was falling. From this, simple thing this fellow did, increase the size of the style, okay. All this plant can do, okay, because plant is the one who is regulating. Did this, from this to this, simple, chalak plant. He is like, now you tell me how pollen grains will come against the gravity. So, gone, okay. First step, successful, students. Second step is self-incompatibility. That means see, I told you that the pollen grain will fall, okay. The pollen grains are going to fall, but it is up to the stigma to choose it or not. Like, are you right for me? I will choose, right? You cannot tell me I am right for you, I am right for you. If you tell, I will not listen. I should feel that you are right for me. So same way the stigma will be like, I should feel you are right for me. So the stigma has got the stigma has understood that I am going to reject all the pollens who are from the same plant, okay. So self-incompatibility, rejecting the pollen, self-pollens, okay, rejecting the self-pollens. This way what will happen, students? Self-pollination can be avoided. Then the third step is changing the receptivity of the pollen and the stigma, okay. Receptivity, that means maturation period. This I am writing for your knowledge, like for you to understand easily, okay. Maturation period, that means anther and stigma, if both of them are maturing at the same time, then there is problem. So what did this fellow do? He told anther, you mature later, stigma, you mature first. Now you tell me how these two people are going to do self-pollination. Very chalak, no, students. So this also is done, receptivity also is done. Now no way they are going to come together, they are not going to pollinate together. Last number was forming unisexual flowers. Forming what? Unisexual flowers, where there is neither only Androecium or only Gynoecium, stamen or carpel. Clear? Yeah, what the Sahil is doing, he is telling full map. Is this clear? Tell fast. The plant can decide, Selvi, if they want to form unisexual, like they will do like all the hormonal changes will be according to that, okay. So there are 4 outbreeding devices, understood? 4 outbreeding devices. We can do artificial hybridization also, that is your emasculation and bagging, okay. That is done with the help of humans, right. They will only remove the anther, they will remove the pollen grain, so that, and they will cover the stigma, so that whatever we want we can add it into that. Got it? Yeah, plants are chalak, yaar, very chalak, but not as much as you people. You people are more chalak. Done, done, done. Okay, next topic, pollen-pistil interaction. Ready? Yes or no? Okay, 2 minutes I'll sit, students. My back is hurting. Okay, just 2 minutes. You people breathe by then. Now we have only 2 more very important topics. After that, all are small topics, okay, which you have studied in morphology. So we'll go through NCERT, okay. How is my botany class who are attending for the first time? Tell me, how is my botany class who are attending for the first time? Some of you have attended many classes, I know. Okay, tell me, how is it? And have we reached the target? Let me check. I cannot see here. Let's say people are escaping. Okay, we are close. We are close. Haha. Satvik, what does that mean, da? What does that mean? I want explanation. There's nothing like that, students. Sir will teach human reproduction. I'll teach sexual reproduction in flowering plants. Okay, next year we'll again exchange. So it's like that. Sometimes we get bored, no? Of our, so we'll take like this plants. Oh, wow, Rathis, what a dialogue. Okay, he's like that, this people, democratic, diplomatic, something, some other word also is there. Okay, he's like that. He will not touch that side, he will not touch this side, he will play a safe dialogue. Okay, you're playing Big Boss, no, in the class. Yeah, you should tell like Madhumati, you should tell like Jasmine, you should tell like Sukanya. Those are my real students, others all. Oh. Okay, chalo. Exactly, Sony scold him. Come on, let's do pollen-pistil interaction. So students, pollen-pistil interaction is very important for you to understand double fertilization also, triple fusion also, because all this is going to be continuation. Okay, all this is going to be continuation. Now, look here, as I told you, stigma has the right to decide, ki, are you the right one or are you the wrong one? Okay, are you the right one or are you the wrong one? So, when, see, we have to draw a diagram, then only we will be able to understand. So, this is the stigma, okay, this is the style, and this is the ovary. This is the ovary. Now, when pollen grains fall on the stigma, okay, when pollen grains fall on the stigma, let's say one pollen grain fell. Okay, one more pollen grain fell, and one more pollen grain fell, because all this is coming by wind, insects, any pollen can come in, no, any pollen can come in. So many pollens have come in. Now this is the embryo sac inside. Okay, this is the embryo sac. Inside this embryo sac again antipodals are there. Okay, synergids are going to be there. Okay, our egg is going to be there, and most important, filiform apparatus will be there, and here polar nuclei will be there. Now let's assume, students, these people are all 1 boys. Okay, if you want, we can pick names from our class itself. Okay, this is Satvik, this is Rathis, this is, who wants to become one more? You know, sometimes I'm scared to call these people's name. Some name and all they'll put, and I'll think that is a boy. They'll be like, no ma'am, I'm a girl. I'll be like, why did you put that name then? One more boy we need. Who is the one more boy? Sahil. Okay, Sahil. 3 boys are there. Okay. Now let's imagine one is doctor, one is engineer, one is bank manager. So one is going to give an offer for the stigma. Now the stigma has to understand who is compatible, no? So let's remove this boy's name, it's too difficult to remember. So we'll just think 3 pollen grains. So first pollen grain is going to be like, you know what, I'm going to take care of your family, I'm going to take care of all of the health, I'm going to take care of everything, so you please accept me, I am the right one for you. Then the bank manager will tell, how many ever loans you want, I will give you the loans, you take your parents to Malaysia, Singapore, anywhere you take them, I will give you all the loans. Another person will tell, you know what, I am a business person, so I am going to make a lot of money. Maybe I am busy right now, but later I will be able to give you time after my 40s, okay? So you should definitely choose me as the right person. So these 3 will have first an interaction with the stigma. So I am not telling this for joke, okay? You should understand that this is exactly how it happens in the plants also. How in our lives all these things happen, same thing happens here. First is going to be, students, they are going to share something called as chemical dialogues. What are they going to share? Chemical dialogues, or we can also say chemical interactions, okay? Dialogues, interaction word also is given in your NCERT. So you can say interactions happen, chemical interactions, that is giving promises that I will do this, I will do this, I will do that. Now the stigma's choice it is, okay? Stigma's choice it is ki, do I have to select this person or do I have to select the other person? Now after the chemical dialogue or after the interaction, the pollen is going to accept, okay? Pollen is going to accept. So the symbol of acceptance is that the pollen tube formation, okay? Pollen tube formation. So let's say our bank manager got accepted, okay? Bank manager got accepted and the pollen tube formation started. Pollen tube formation started. The other 2 got rejected, okay? Other 2 got rejected. So the first, okay? First step was chemical interaction where they spoke to each other. The second step was pollen-pistil, okay? Pollen-pistil, we can tell pollen-pistil interaction, okay? That and chemical dialogue will be the same. So I will just write here pollen tube, okay? I'll directly go to this, pollen tube germination, okay? Pollen tube germination. That means interaction over, acceptance happened, okay? Interaction over, accept, they remember whose name I told, my god. Acceptance has happened. So here germination is going to happen. Students, usually when germination happens, there is a need for boron, okay? Just remember that. If you can remember, well and good. If not, it's okay, okay? There is a need for boron, that do you remember. Now students, after acceptance, pollen tube will not stop there. Pollen tube is going to continue, yes or no? So the pollen tube is going to continue the formation and it is going to extend all the way like this, okay? So it is going to extend, it is going to extend up to where? Up to the embryo sac it is going to grow, okay? Now I am drawing it outside because I do not have space. So don't think that ma'am this is going outside, it's going inside only, okay? It's going inside only. Is this clear? So third thing is going to be the continuation of the pollen tube formation, okay? Here germination. So let's write like this. Let's write this step as germination, okay? Let's write this step as germination. Okay, third step will be pollen tube formation, okay? Third step is pollen tube formation. Okay, pollen tube formation. Now, listen to me carefully, okay? Listen to me carefully. Now, before it is entering the embryo sac, this will go through the formation of the, what is this, pollen grains, no? They have the cells, no? The male gamete and the vegetative cells. So, they will think, most of them, majority of them, that means at a 60% of them, in the 2-cell stage, shedding is going to happen. We have heard, no? 2 cells shedding is going to happen. That means one generative cell is going to be there, one vegetative cell, one generative cell is going to be there. That male gamete will go and fuse with the egg, kel khatam. Okay, but in some of them, what is going to happen? 40%, in the 40% students, 3-cell stage is going to happen, okay? So, in 40%, 3-cell stage is going to happen. That time what is going to happen? This After this pollen tube formation, if it is a 2-cell stage, 1 vegetative cell, 1 generative cell. If it is a 3-cell stage, they will undergo mitotic division and they will form 1 vegetative cell and 2 generative cell. 2 generative cell. So, that means this will give us 1 male gamete, okay, 1 more male gamete. 2 male gametes they are going to give. But we are expecting only 1, but 2 male gametes, okay. 2 male gametes are going to be there. Yes or no? Now, students, what to do? Problem will happen, no? We expected only 1 marriage to happen, but 2 marriages are going to happen. So what is going to happen? Yes. So, what is going to happen, students? At the 3-cell stage, one fellow with the help of filiform apparatus is going to fuse with the egg. Okay, one fellow is going to fuse with the egg. Another fellow will go and fuse with the polar nuclei. What is going to happen? One fellow, so listen to me carefully, I'm teaching you double fertilization, triple fusion and pollen-pistil interaction together. Okay, we always teach it together because if you teach it separately, it's too difficult to understand. So, at a stretch you understand that pollens will fall, the right compactable one, okay, the compactable pollen will start germination. This much only you have to know here, okay. The right type will continue the germination, the wrong type will be rejected, then and there it will be rejected, okay. Second thing is, if it is in a 2-cell stage, then we are going to have fertilization happening. That means one gamete will go and fuse with the egg, we will get the embryo, sorted, story over. But if it is in the 3-cell stage, what is going to happen? In a 3-cell stage, students, okay, in a 3-cell stage, sorry, one second. In a 3-cell stage, one vegetative cell that will completely degenerate, okay. Why? Because providing all the goodness for this generative cell to grow, that fellow will go off. So, one male gamete, listen to me carefully, this is your triple fusion and double fertilization. One male gamete will fuse with the egg, okay, and form what? Form the main hero, embryo. Form embryo, okay. Second scenario, okay, scenario, the second male gamete, okay, the second male gamete will fuse with what did I tell you? With the polar nuclei, with the polar nuclei. So, students, this is the embryo sac. This embryo sac is what? 8 nucleated 7-cell stage. 8 nucleated 7-celled stage. Why do we call it like this? Because it's very simple. 1, 2, 3, 4, 5, 6, 7, 8, 9. Sorry, what counting is this? 1, 2, 3, 4, 5, 6, 7, 8. Okay, 8 nucleus is there, right? 8 nucleus is there. But 7 cell is there because we are going to consider this polar nuclei as one cell. Polar nuclei as one cell. Yes or no? Clear? Thank you. Okay, one cell. So, 2 male gametes plus 1 polar nuclei is going to give me the PEN, that is primary endosperm nucleus, or we can chuma write endosperm. Okay, we can write endosperm. Any doubts? Any doubts? If you understand this, then this chapter is easy peasy, students. We have done with all the important topics. If you understand this one concept, this is where question will come. So, you should understand this thoroughly. Ask me doubt, I will explain again. Sorted? Sorted? What is going to happen? What is going to happen? One male gamete will fuse with the egg. That was the actual plan. Only one marriage we thought, elder brother, elder sister, over story. But in that, younger brother also came. He's like, even I want to get married, please, please, please. I met her sister, even I want to get married. Then we are like, okay, fine, you also get married. Fine. That will become what? So, this is called as double fertilization because 2 fertilizations are happening. Okay, this is only going to happen in a 3-cell stage. Okay, double fertilization happened. This is called as triple fusion. Why? Because this is going to form a triploid. This is going to form a triploid because students, this is 2 nucleus, 1 cell. 2 nucleated, 1 cell it is. So, this is going to be a triple fusion. Is this clear to all of you? Can everyone please answer? Okay, I am asking from that time. All of you please answer fast. Students, I am writing all this small things also for you people. Okay, so make sure you download the notes from Vedantu NEET English or from Biology by Gopika. Okay, pakka clear? All of you? Thank you guys. Thank you so much. Okay, done? This is also clear, no? What I wrote here? This happens in your 3-cell stage. Okay, where your double fertilization and your triple fusion happens. Triple fusion, double fertilization and triple fusion happens. This is your whole pollen-pistil interaction. First one is going to be chemical dialogue, second is going to be germination, then the pollen tube formation with the help of filiform apparatus. Do not forget the role of filiform apparatus. Filiform apparatus is going to be the one who is going to lead till the egg. Okay, till the egg. Any doubts so far? I hope it's clear. Okay, I hope it's clear. Chalo. I think here I have left more space. Students, I thought I will write a lot, but no, don't need to write that much. Okay. Yes. So, look here, this is the diagram. Now, look at the diagram. Now that you understood the concept, try to think of the diagram. See students, here the plasma membrane is there, synergids are, filiform apparatus is there. See, can you see the pollen grain is entering? So, there is the vegetative cell and 2 male gametes. Okay, this is 2 male gametes. So, the generative cell, after it has come down a little, it went through a mitotic division. Because of that mitotic division, it gave rise to 2 male gametes. That 2 male gametes, through the filiform apparatus, it is entering. Okay, and one will go and fuse with the egg and one will go and fuse with the polar nuclei. So, here you can see male gametes. One male gamete has gone here, one more male gamete is here. That means this fellow will fuse with the polar nuclei, this fellow will fuse with the egg. Okay, this is going to give rise to the rise of pen, that is your primary endosperm nucleus, or you can remember endosperm, and this is going to give rise to the embryo. Clear or not? Okay, this is going to give rise to the embryo. Easy, no? Very easy, no? You might have studied it before and you would have found it very difficult, but it's not very difficult, it's very easy. Okay, so we'll just write that, students. So, double fertilization means where one male gamete, okay, one male gamete is fusing with egg, okay, to form embryo, okay, first fertilization. Second one again, okay, second situation is happening when one male gamete is fusing with the polar nuclei to form primary endosperm nuclei, or simply you can write endosperm. And this endosperm is going to be the one that is going to provide the nourishment for the growing embryo. Okay. Thank you, Anushya. I think your pronunciation is correct. Okay, students, this endosperm is going to be the one who is going to provide the nourishment for the growing embryo. Chuma, they won't make anything, okay? It's going to provide the nourishment for growing embryo. Got it? Clear? All of you? Yes. Now, I will tell you one more thing. So, this process is called as syngamy. Okay, this is called as syngamy. This is going to give rise to your diploid, normal thing, no? This is going to be, okay, this is going to be triple fusion. Okay, this is going to be triple fusion, and this is going to give rise to triploid. Okay, triploid. This was also clear, right? Yes, okay. Now, what is double fertilization? Double fertilization is going to be nothing but, okay, double fertilization is going to be syngamy plus triple fusion. No one is in office, students. I am getting scared when I am hearing one sound. Wait, let me check. Who is that? Okay, no one is there, I guess. Okay. Clear? Everything is clear. Double fertilization, pollen-pistil interaction, triple fusion. Ploidy is also clear. Any doubts in ploidy? Ask me now. Ploidy should be clear. 2n and 3n. Okay, ploidy is. Okay, clear, right? Wow, let's see if we'll reach the target today. Done. Done. Okay. Now, last topic is, students, not last topic, artificial hybridization. Okay. Artificial hybridization is very easy. Two steps are there: bagging and emasculation. That means I want to artificially select what I want to, fuse, or what fertilization should happen. Because if I let it for cross pollination, definitely cross pollination will happen. Okay, it will happen. But the thing is that I need to make sure that I am going to get a high variety, a good yielding plant, or this flower I need, because okay, something like that. So, for that I am going to do artificial hybridization. I will remove the anther. Okay, that process of removal of the anther is called emasculation. Listen to me carefully. Removal of anther is called emasculation. Then I will do bagging of the plant. That means I am going to bag the stigma so that unnecessary pollen grains from other plant does not come and fall. Then I will take the pollen grain that I want and I am going to dust it on the stigma and again bag it. That is called rebagging. This is artificial hybridization. Okay, so look here. As you learn in the earlier chapters on plant breeding, chapter 9, a breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. So, artificial hybridization, okay, artificial hybridization is one of the major approaches of crop improvement program. Okay, crop improvement program. Got it? In such crossing experiments, it is important to make sure that only the desired pollen, okay, only the desired pollen grains are used for pollination and the stigma is protected from contamination, from unwanted pollen. So, this is achieved by emasculation and bagging. So, look here, students, if the female parent bears bisexual flowers, removal of anther, okay, this is going to happen in bisexual flower. In unisexual flower, such a problem does not exist. Why will I take headache of whatever is not there? Okay, so removal of anther from the flower bud before the anther dehiscent, okay, dehiscent with a pair of, let's say, forceps, okay, with a pair of forceps, is going to be called as emasculation. Okay, it's going to be called as what? Emasculation. Emasculated flowers have to be covered with a bag. Why it has to be covered with a bag? Guys, come back to the class. What are you people talking about? Okay, it has to come back, sorry, come back to the class it seems. Okay, emasculation, after emasculation I have to make sure bagging is happening, otherwise some other pollen will come and fall. Okay, so it has to be covered with a suitable size, and we usually use butter paper. Okay, butter paper to prevent contamination. To prevent contamination of the stigma with unwanted pollen. Okay, stigma with unwanted pollen, and this process is called bagging. Isn't it easy, students? Isn't it easy? Yes or no? When the stigma of the bagged flower attains receptivity, mature pollen grains will be collected and dusted on the stigma, again rebagged. Okay, the flowers are rebagged and the fruits are allowed to develop. Clear? Post fertilization events we are going to start. Okay, post fertilization events we are going to start. Okay, chalo. So, pre-fertilization events, fertilization events are clear. Under fertilization, you have to study double fertilization, triple fusion, pollen-pistil interaction is a part of it. Okay, in the pre-fertilization event, microsporogenesis, megasporogenesis, pollination. Are we good to go? Can we start post-fertilization events? From here, I have used NCERT SNP students because it's all topic that you have studied in morphology. So, I am just going to recap. Okay, I am just going to go through the border and I will tell you this is what you have to understand. Okay. So, this is going to be from NCERT. So, chalo, let's start. Post fertilization events. I told you post fertilization events are nothing but ovule becoming seed, fate of these people. Okay, ovule becoming seed. Okay, ovary becoming fruit. You can recap your morphology as well. Embryo, endosperm. This is all that we have to learn. Okay, and some exceptions also. Some exceptions also. Okay. Now, endosperm development precedes embryo development. Why? Students, they are asking why. Answer. The primary, see, endosperm development will be first. So, development of embryo will happen after the development of endosperm. Be the globular heart plus suspensor, be the heart-shaped embryo plus the suspensor forming the mature embryo. All of this will happen after the endosperm develops. Why? Why? I want the answer. Let's see who is the smart one in the class today. And I wish I could come and give you chocolate, but I cannot. Who is going to tell me why endosperm precedes embryo? Something I had told you when I was teaching you triple fusion. Think and tell. It's very easy, very easy. Let's see, let's see. A power play. What are you telling? Tell fast. Jaldi bolo. No one, no smart kids in the class, sir. That will keep happening with that, students. If I ask question, they will decrease and go off. Very good, Shakti, Shakti, very good, very good. I want all of you to clap for Shakti. Okay, Shakti is the only student who has answered. Students, it's so simple. If endosperm is not developed first, how will the embryo get the nourishment? Who is going to give the nourishment? Right? For the nourishment to be provided, I need endosperm. Before making food only, I am sitting and telling, oh, I feel like, oh, I should. First, food should be formed for me to go and eat. No, same thing. Endosperm should be formed so that the growing embryo gets the nourishment. Okay, all claps for Shakti. Come on, my students are very encouraging. They'll clap. See, I told you, no, they're very encouraging. They'll clap. So, endosperm development precedes embryo development. Why? The primary endosperm cells divide repeatedly and forms a triploid endosperm tissue. The cells of these tissues are filled with reserve food materials. Okay, reserve food materials. You didn't understand? Very simple. What is endosperm's job? After triple fusion, when we form the endosperm, what did I tell you its job was? To provide nourishment for the growing embryo. Yes or no? If embryo does form first, how will you provide nourishment? Right? So, first endosperm should be formed, then embryo should be formed. Is this clear, Tanuja? First you will make the food, then only you will eat it. No? Before making the food only you will start eating. Simple like that. Okay? First you should study to write the exam. So like that, everything interrelated. So that is what it is. Clear? Okay, got it. Now, so are used for nutrition for the developing embryo. See, this is the reason why it should precede. Okay, why it should precede. In the most common type of endosperm development, the pen undergoes, so pen is primary endosperm nucleus, will undergo successive nuclear divisions. Okay, it will undergo successive nuclear divisions to give rise to free nuclei. So students, here all free nuclear divisions are going to happen. Okay, free nuclear divisions are going to happen. Now, this stage of endosperm development is called free nuclear endosperm. So, free nuclear endosperm, cellular endosperm and all we tell. Free nuclear endosperm means what? Continuous nuclear division is happening. Okay, no walls, no formation is happening. Only nuclear division is happening. That means from 2 nucleus to 2 nucleus to 4 nuclei, 4 nucleus to 6, 6 to 8. That means what? This is a free nuclear division. So, it's a free nuclear endosperm. Okay, this much will you understand, don't break your head and all. Free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. That means till now the nucleus was forming. So, I called it free nuclear division. Slowly cell wall started forming. So, I will call it cellular. Okay, I will call it cellular. Anyone anyone has a doubt? See students, very simple. Let's say at the first situation, okay, at the first situation, this is your embryo. First it was 2 cell stage. Okay, first it was 2 cell stage. From 2 cell stage, okay, it became 4 cell stage. Okay, then from 4 cell stage, this I am just drawing to make you understand. Okay, actually you should draw cells, so many cells. They became 8 celled. Okay, they became 8 cell stage. This was free nuclear division where the nuclei was continuously dividing. In the second scenario, okay, this is free nuclear division. In the second situation, what happened? This was happening, okay. Slowly cell wall formation started happening. Okay, cell wall formation started happening. This became a cellular division. Okay, I cannot draw the exact image, it's very complicated to draw. So, cellular division was happening. Is this clear? Just to make you understand, I am drawing. Fine? Chalo. So, who was doing this division? Pen is doing the division, okay. That is primary endosperm nucleus will keep going through successive divisions and it will become free nuclear endosperm. Okay, free nuclear endosperm. This free nuclear endosperm, when cell wall starts forming, okay, cell wall formation happens, it will become cellular endosperm. This much you understand, please don't complicate anything, just understand this much. Can you all think of this much? Make a note of that, what I have written in white color quickly. What I have written in white color, I want all of you to remember write quickly. Students, I am sitting 2 minutes, okay, because my back is paining. Don't think where is ma'am going. Just write that white color thing is very, very important. Oops. Okay, let's do one thing. In this only I'll draw one, students, so you'll be able to understand. Yeah, look here. I'm going to make a better diagram for you people. Now let's say here only cell formation happened. Okay, cell formation happened like this. Okay, like this cell formation started happening. So this becomes your cellular division. Can you relate now? Can you relate? Yeah, coconut water. Yes, yes, we are going to come to that. Okay, students, just give me 1 minute. I'll just draw 2 diagrams and I'll make you understand the cellular formation. I feel because this diagram and that embryo sac diagram look similar. Later when I share the notes, no, students will be like, oh God, what is this? We have never learned this. So that's why I'm just drawing oval here. So I'll tell you, same thing students, similar thing what we just learned now. Let's say this is your nuclear divisions, okay, nuclear divisions. many nuclear divisions was happening, okay. Few were here, okay, that became this much. Now in the next stage, what is going to happen? Here cell formation happened, okay. Slowly cell formation started happening, cell wall formation, understood. So this cell wall formation led to the free, so from here, let us say this was the free nuclear division, okay, free nuclear division. From here it started forming cell wall, okay, cell wall formation. Is this clear? Now it's better? You are able to understand? Yes, no, cell wall formation. Chalo. Now that is clear. So let's go back here. The number of free nuclei formed before cellularization varies greatly. The coconut water, the coconut water from the tender coconut, okay, the coconut water that we drink, no, from the tender coconut that you are familiar with is nothing but the free nuclear endosperm, okay, free nuclear endosperm made up of thousands of nuclei. So that coconut water has gone through free nuclei, okay. Will all of you comment today? Okay, day 4 Navratri series done? Yes or no? Okay, free nuclear endosperm made up of thousands of nuclei, okay, thousands of nuclei surrounding white kernel is going to be the cellular endosperm. So once the cell wall formation happens, it is going to form the cellular endosperm, okay. Chalo, done. Example also clear? Clear, all of you? Now, embryo develops at the micropylar end of the embryo sac where the zygotes, okay. Most zygotes divide only after certain amount of endosperm is formed because they want the nourishment, okay. Now, this is an adaptation to provide assured nutrition. See, this is your answer. This is what I was expecting. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocots as well as dicots. All this you have studied in morphology, so we are going to shh. Okay, yes, sure, okay, okay. Oh, it's 9, 9, 10, oh my god, okay. So yes, so students, this is all what you have studied in morphology, so we will do a quick recap. Are similar in both monocots and dicots. Now, the zygote gives rise to pro-embryo and subsequently globular, heart-shaped and mature embryo, okay. So here, 1 second students, where is the diagram? Oh no, I didn't put the diagram, okay. Never mind. See, just understand 3 stages are there, okay. First is going to be pro-embryo, okay. Next is going to be globular embryo. Students, all of you can open your NCERT, there is a diagram given in you, okay, pro-embryo, understood. Then heart-shaped embryo, okay, heart-shaped, and then matured embryo, matured embryo. Yes, if Tanuja tries, she can be, okay, but she has to try. She sits all the classes till the end, so it's definitely one of our loyal students, so we wish her all that. Yes, globular, Tanuja is there, Soni, Madhumati, Ratas, all these are our regular students, okay. Did I miss someone? Yeah, Shu. So globular, heart-shaped and mature embryo, these are 3 things, okay. Starts with globular plus you can see suspensor, okay, suspensor, then heart-shaped plus suspensor, okay, finally forming the mature. In the mature, what and all is going to be there? Plumule, your radicals, scutellum, okay, coleoptile, all of that is going to be there. This is the flow chart, okay, this is the flow chart. This is how maturation is going to happen. See here students, same thing. A typical dicotyledon embryo consists of embryonal axis, okay, there is going to be an embryonal axis. Embryonal axis is nothing but, let's just look here, this is your embryonal axis, these are your cotyledons, okay. Whatever is going to go up, it's going to form the shoot, okay, shoot apical. Whatever is down is going to form the root. So this can also be called as the radicle, this can also be called as the plumule, okay. Now 2 cotyledons are there, okay, 2 cotyledons are there. The portion of the embryonal axis above the level of cotyledon, this is epicotyle, which terminates within the plumule, understood. Whatever is above this epicotyle, whatever is below this hypocotyle, this is going to form the plumule, this is going to form the radicle. Plumule is going to form the shoot, radicle is going to form the root. Yes. Okay, chalo. Now the cylindrical portion below the level of cotyledons, okay, cylindrical portion below the level of cotyledon is the hypocotyle. Okay, here it is a plumule. At its lower end is the radicle or the root tip. Okay, radicle or the root tip. The root tip is covered with root cap. Why? Because it has to emerge into the soil. Okay, soil will be hard, so it is going to emerge into the soil. Students, this you know, right? Plumule giving rise to shoot. Cotyledons to monocot or dicot. Okay, hypocotyle again root. Radicle forms the root. Root cap, the portion at the end of the root to help them get inside the soil. Now, coleoptile is the region that is going to help your plumule. Okay, upper portion, no, to protect the plumule, coleoptile will be there. There is coleorhiza at the end of the root, I mean below the root, which is going to protect the root. Okay, see here, coleorhiza is there, no. This you have studied in morphology if I am not wrong. Coleorhiza, coleoptile. Coleoptile is going to protect the shoot apex and coleorhiza is going to protect the root. Are you all listening? See here only it is there. Embryos of monocotyledon possesses only one cotyledon. In the grass family, the cotyledon is called scutellum. So, scutellum is going to give the separation between the endosperm and the embryo. I don't know if you people know this. It is going to separate the endosperm and embryo. Okay, endosperm and embryo. Done? Chalo. Now, that is situated towards one side of the embryal axis. At its lower end, the embryonal axis has a radicle and a root cap enclosed in an undifferentiated sheath called coleorhiza. So, coleorhiza will protect the root portion. The portion of the embryonal axis above the level of attachment of the scutellum is called as the epicotyle. So, just remember this, only this diagram you remember in your head, everything is going to be easy. This is the axis, okay, this is the embryonal axis, this is your cotyledon, whatever is going above, epi, okay, whatever is going below, hypo. Epi will go to plumule, that will form the shoot. Okay, this will go to radicle, that will form the root. Ho gaya? Yes or no? Easy peasy lemon squeezy? Yes? And then what is going to protect the shoot, students? Coleoptile. What is going to protect the shoot? Coleoptile. Okay. Now, two types of seeds are there. I will just write it there instead of reading it. Two types of seeds are there. They are called as albuminous okay, and exalbuminous. Okay, before the board gets stuck, I have to finish writing, students. Albuminous means no endosperm will be left. Okay, non-albuminous means no residual endosperm. Okay, no residual endosperm. That means all the endosperm will be utilized by the growing embryo and these people will be left with no endosperm. Okay, these people will be left with no endosperm. Okay, no residual endosperm. Example is going to be your pea and groundnut. Okay, pea and groundnut. Same thing here, students, exalbuminous, or we can write albuminous only. That means here your endosperm will be left. Okay, endosperm residue, okay, or let's write this way, retains a part of the endosperm, retains a part of the endosperm. Okay, what is the example? Wheat, maize, okay, all of these are examples. Got it, all of you? Yes, wheat, maize, all of this is the example. Done. Barley, anything, all this you can write. Okay, exceptions these are. Non-albuminous and albuminous. Okay, then see, same thing is given here. Students, one more I will write here, this one. okay, when the nucellus, okay, when the nucellus is going to be persistence, okay, persistence, when the nucellus is going to be persistent nucellus, nucellus, we call it perisperm. What do we call it? Perisperm. That means the nucellus is still remaining, student. Nucellus was supposed to give nourishment for the growing embryo sac, but that fellow remained. So in that cases, this will be called as perisperm. An example is going to be beet square, okay, beet and black pepper, beet square, okay, beet square is the way to remember it, okay, done. No mnemonics, yaar, no mnemonics I made. Okay, done. Next one. Here, so far good. Now, integuments of ovules harden as tough protective seed coats, the micropyle. So, see what happened to each of the layer. So, these integuments are going to form the testa and tegmen. Okay, testa outer layer, outer integument, tegmen inner layer. Okay, testa and tegmen. Got it? Of the ovules harden as tough protective seed. The micropylar end, see the micropylar remains as a small pore in the seed coat, and this facilitates the, micropyle, no, I think I told by mistakenly hilum there, okay. So, just make the change. This facilitates the entry of oxygen and water in the seed, okay. Students, there by mistake I told hilum instead of micropyle, okay. As the seed matures, its water content is reduced and the seed becomes relatively dry, that is 10 to 15% moisture by mass. The general metabolic activity of embryo slows down. The embryo may enter a state of inactive called as dormancy. Okay, inactivity called dormancy. That means because the condition is not favorable, the seed will be like, I don't want to actually be active, I will just sleep. Okay, when it goes to sleep, that is called as dormancy. Okay, that during dormancy also your hormones are going to work, your ethylene, auxin, gibberellin, all of this people are going to work, you know that, right? So, or if favorable conditions are available like moisture, oxygen, suitable temperature, they germinate. As ovules mature into seed, so this is the ultimate, ovules will become seeds, ovary will become fruit, okay, ovules into seed, ovary into fruit proceeds simultaneously. That means, students, both of this process is going to happen at the same time. Simultaneously is going to happen. It's not like first you form the seed, then I'll form the fruit. No, simultaneously it's going to happen. Okay, develop the walls of the fruit, and it is, students, the walls of the ovary. The wall of the ovary develops into wall of the fruit, which is called as the pericarp. So even the wall of the ovary has a job to become the pericarp. To become the pericarp. The fruits may be fleshy as in guava, orange, mango, or guava. Okay, in most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. I told you, know, whatever is not required, it will degenerate and fall off. However, in a few species such as apple, strawberry, cashews, they are false fruit. That means from ovary, fruit did not develop. From thalamus, from other parts, the fruits have been developed. Okay. So, degenerate. However, in a few species like apple, the thalamus also contributes to the fruit formation. Is this clear? So, that kind of fruits, what will I call it as? False fruits. Whom will I call true fruits? The one who are developed from the ovary. Okay, who are developed from the ovary. So, only two more concepts, polyembryony, apomixis. So, we will just write about the fruits. Okay. Okay, chalo. Different types of fruits. Another 10 minutes, we should be done. Okay. First category is going to be true fruits and false fruits. Okay. True fruits, false fruits. Yes or no? Chalo. Now, remember, true fruits are going to be the ones which are developing from the ovary. Okay. Developing from ovary. Okay, example, you can write mango, anything you write. Okay, example. False fruit, developing, you can write developing from thalamus, okay, as given in your NCERT. Example, apple. Okay. Now, next category is going to be called as parthenocarpic fruit. What fruit, students? parthenocarpic fruits. That means this fellow developed without even fertilization. Okay, this fellow developed without fertilization. Understood? Development without fertilization. Example, banana. Got it? Then, polyembryony is there, apomixis is there. Okay, polyembryony is there, apomixis is there. So, apomixis students, I will continue here earlier, all the terms, so it will be easy for you. Apomixis is when seed develops without fertilization. Seed develops without fertilization. Clear? Students, fruit without fertilization, parthenocarpy, seed without fertilization, okay, is called as apomixis. Example is going to be citrus, mango, okay, all these people are going to come under this one. Citrus and mango. Clear? Seed without fertilization, fruit without fertilization. What did I write here? Yeah, I will write here fruit without fertilization. Okay. Okay, fruit without fertilization, banana, seed without fertilization, apomixis. Now, last one is going to be polyembryony. What is polyembryony, students? From the name only we know that many embryos are formed. Okay, many embryos are formed like in citrus. Okay, thank you Ashwati. Okay, occurrence of more than one embryo. Okay, occurrence of more than one embryo. Example again, citrus fruits only. Okay. Okay, done, 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 done. Okay, target also reached, chapter also done. Today my only thing was that I was like, today you see my students will do it. Okay, I'm just going to check, okay, whatever, did we do everything? Polyembryony done, apomixis. Okay, here, although in general are products of few flowering plants such as Asteraceae grasses have evolved in such mechanism to produce seeds without fertilization. Okay, here see apomixis. Okay, what fruits? So that is parthenocarpy. Students, apomixis, parthenocarpy, don't get confused. Apomixis, what is going to happen? It is going to mimic sexual reproduction. Okay, it's going to mimic sexual reproduction. Clear? Yes or no? So what is going to happen? The diploid egg cell is formed without reduction division and it will develop into embryo without fertilization. So when it develops into directly embryo, then what is the tension? Okay, so this is how apomixis is going to happen. It happens in many citrus plants and mango, as I already told you. Polyembryony, what happens? Occurs of more than one embryo. Okay, more than embryo seed is called as polyembryony. This can happen, students. What happens? See, nuclear cell surrounding the embryo sac starts dividing. That could be the reason. Okay, protrudes into embryo sac and develop into embryos, because nucleus has the reserve material. What it will do, it will start dividing and it will get into the embryo. So many embryos will be formed. Okay, many embryos will be formed. This is called as polyembryony. Okay, yes, done. Done. Will you do a homework question in the comments? I'm giving you the first homework question. Too difficult to type, huh? No, right? Okay, do this. The cells of which of the following layers of the anther walls are multinucleated due to endomitosis? Multinucleated. Okay, this is your homework. Homework in the comments. Students, that hilum thing, just remember, okay? By mistake, I told hilum instead of micropyle. Homework in the comments. This is the homework question. Okay, which is the cell that will have multinucleated because of endomitosis? You already know the answer. I told you multinucleated multiple times. You know the homework. So I want it in the comments, and also let me know how the session was. Okay, how was Navratri series. Again, you will meet me only after 2-3 days. Okay, so by then you keep commenting. How was Navratri series day 4? Okay, and how is my botany class for the people who joined first? So yes, thank you so much students for joining. I hope all of you enjoyed the session. I know it was a long session, but definitely the session was definitely helpful for all of you, I believe. So thank you so much for joining. See you all in the next Navratri series, which I'll be taking on Thursday. So do not fail to join. I'll be taking evolution chapter, which I'm going to do as a one-shot class. So it is like a story telling that entire chapter. So we'll be having so much fun in the chapter. So see you all in my next Navratri series that is going to happen on Thursday. That will be evolution chapter. Okay. So I want all of you to take some times and comment and let me know, because students, all of us are standing for this many hours, and I want all of you to respect what you have right now, because once it's gone, it's actually very difficult to come back, right? Now that you have it, you might not know the value, but once it's gone, you might know the value. So definitely appreciate your teachers who are standing here and teaching you for free, and we want all of you to do well in your exams. So try to do as much as possible by yourself. And yes, thank you so much. After I reach home, I'll definitely post it on Telegram. Okay, and I will share the notes as well on the Telegram, that is Biology by Gopika and Vedantu NEET English Telegram. Do join the Telegram and do join our WhatsApp channel again with the same name. So see you all next time. Thank you so much. Those who are joined new, I hope you really love the session. Do not forget to subscribe to the channel and smash the like button. And students, let me tell you, you have a chance to win your prizes. That is, your MVSAT exams are still happening. You can take the scholarship test and you can get a chance to win iPhones, PS5s and drones and other things. This is for the students who joined newly. Okay, for those students, MVSAT exam registrations are available. So definitely take 2 minutes, go to the description box. Students, go to the description box here and click on the first links. Okay, once you click on the first link, you will be able to know the test timing. See, it is starting from 10th to 20th October online. Okay, up to 100% scholarship. And students, here it is going to start it. It has already started in the offline centers. So definitely write the MVSAT exam and get an opportunity to know your strength, know your talent. If it is not about the prize, forget about the prize, but at least to know where you stand, definitely do this. Thank you so much. See you all. Tata, bye-bye.